The reef at low tide feels less like open ocean and more like an exposed city of stone. Water thins into warm, wavering sheets across coral rubble and sand channels, leaving tide pools scattered between knotted outcrops of limestone and dead coral skeletons. Small fish weave between shadows, and crabs test the air with cautious movements. To an untrained eye, this terrain appears static and mineral, an uneven scatter of rock and algae. Yet in the quiet depressions where sand settles and filamentous algae wave lazily, a form lies so perfectly integrated into the substrate that it ceases to look biological at all. The stonefish does not announce itself with motion or color. It erases its identity entirely. Its body resembles a fragment of reef broken loose and partially buried. The skin is textured with fleshy lobes and ridges that mimic encrusting algae, sponges, and sediment. Pigmentation varies from mottled browns to dull greens and grayish reds, reflecting the palette of the surrounding habitat. Even the eyes, positioned high on the head, protrude only slightly and blend into the irregular surface. In many individuals, filamentous growths cling to the skin, enhancing the illusion of inert rock. Camouflage here is not merely coloration, but architecture. The outline dissolves into the substrate, eliminating visual cues that would signal predator or prey. Camouflage is the first layer of defense and the primary tool of predation. The stonefish hunts by remaining immobile for extended periods, often partially buried in sand, leaving only the dorsal ridge and eyes exposed. Small fish seeking shelter among coral rubble approach without hesitation. When a target swims within range, the stonefish's mouth expands in a rapid suction strike, creating a pressure differential that pulls the prey inward in a fraction of a second. The action is explosive but brief, and the fish resumes stillness almost immediately. There is no chase, no display of speed across open water. The hunt is decided before the prey realizes that the rock beneath it has a mouth. Yet the stonefish's most formidable adaptation lies not in the mouth, but along the dorsal surface. Thick, stout spines rise from the back like broken shards of coral, each connected to venom glands at its base. These spines are not designed for active stabbing in pursuit of prey. They are positioned for a different scenario entirely. The venom apparatus functions as a pressure-triggered syringe system primed not for attack, but for response. When downward force compresses a spine, a sheath surrounding it is pushed aside, exposing a groove through which venom is forced upward and into whatever has applied the pressure. This mechanism reveals the stonefish's dual strategy. It wins hunts by invisibility, but it survives by punishing intrusion. Reef environments are crowded with larger predators, including groupers, rays, and sharks, that may investigate unfamiliar shapes. More commonly in shallow coastal areas, terrestrial animals and humans wade across tide pools, unaware of what lies beneath their feet. In these contexts, the stonefish cannot flee quickly. Its body is heavy and adapted for stillness rather than sustained swimming. Camouflage delays detection, but if concealment fails, venom becomes the second layer of protection. The distinction between touching and stepping is critical in understanding the stonefish's risk to humans. A cautious hand brushing lightly against the dorsal spines may trigger some venom release, but the force is limited. In contrast, a foot bearing full body weight drives the spines downward with substantial pressure. The greater the compression, the deeper the spine penetrates and the more venom is injected. The spines are robust and can pierce through soft footwear, embedding into tissue before the individual has time to react. In effect, body mass amplifies the venom delivery system, converting the stonefish into a biological landmine triggered by force rather than proximity. The venom itself is complex, containing proteins that disrupt cellular membranes and interfere with cardiovascular and neuromuscular function. In fish predators, envenomation can cause immediate pain and rapid retreat, discouraging further attempts at ingestion. For a predator that might otherwise swallow the stonefish whole, the cost of that attempt becomes prohibitively high. In humans, intense localized pain is often the first symptom, sometimes described as disproportionate to the visible wound. Swelling, tissue damage, and systemic effects can follow depending on the depth and number of spine penetrations. The severity correlates directly with pressure and contact area, again reinforcing the principle that the mechanism is designed around force. Despite its reputation, the stonefish is not aggressive. It does not pursue divers or launch defensive strikes unprovoked. The dorsal spines remain sheathed until compressed. In controlled observations, stonefish often remain motionless, even when closely approached, relying on camouflage rather than intimidation. This passivity underscores the evolutionary logic of the venom system. Energy is conserved by avoiding unnecessary confrontation. The cost of producing venom is balanced by its role as insurance against catastrophic predation or accidental trampling. The reef ecosystem reflects the stonefish's presence in subtle behavioral patterns. Small fish species that have evolved alongside stonefish may exhibit cautious hovering above substrate, avoiding direct contact with irregular surfaces. Some reef inhabitants, such as certain shrimp species, appear capable of recognizing the stonefish's outline and avoiding close proximity. These interactions suggest that camouflage, while highly effective, is not infallible. Over evolutionary time, prey species that can detect minute deviations in texture or shadow may gain survival advantages. In response, stonefish maintain remarkable variation in surface morphology, preventing predators and prey from forming a single reliable search image. Thermoregulation also influences the stonefish's behavior in shallow waters. As ectothermic animals, their body temperature aligns with ambient water conditions. In warm tropical shallows, metabolic rates increase, supporting active digestion following successful ambush. During cooler periods or in deeper waters, activity levels may decrease, extending intervals between feeding events. Because their hunting strategy relies on patience rather than pursuit, fluctuations in temperature primarily affect digestion and venom synthesis, rather than locomotion. The capacity to remain still for prolonged durations reduces energy expenditure and allows survival in habitats where food encounters are sporadic. Juvenile stonefish face particular vulnerability before their camouflage fully develops and before venom glands reach peak potency. In these early stages, mortality rates are likely higher, selecting strongly for rapid development of dorsal spine defenses. The relationship between growth and venom capacity highlights the interplay between morphological and chemical adaptation. A fish that cannot yet fully resemble a rock must rely more heavily on the deterrent effect of its spines. As it matures and camouflage improves, the reliance shifts subtly, though both layers remain active. In the broader toxic food web, the stonefish occupies a defensive apex of sorts. It does not accumulate toxins from algae or synthesize complex venoms for predatory immobilization like other reef organisms. Instead, its venom functions primarily as retaliation insurance. The reef floor becomes safer for the stonefish precisely because stepping on it carries consequences. Predators that learn through painful experience are unlikely to repeat the mistake. This learned avoidance creates a protective halo around individuals, reducing predation pressure not only on the stonefish itself, but potentially altering the foraging routes of larger animals. Observing a stonefish in situ challenges assumptions about danger. It lies inert, blending seamlessly into coral rubble, its presence betrayed only by subtle contours in shifting light. A small fish drifts too close and vanishes into the mouth, an ambush completed with surgical speed. Minutes later, calm resumes. A passing ray glides overhead, careful not to press downward on irregular substrate. The stonefish remains unchanged, its dorsal spines sheathed, its venom undisturbed. The true hazard emerges only when weight descends from above, when camouflage fails to prevent contact. In that instant, the reef's quiet surface transforms into a concealed defense system. The spines depress, venom flows, and the cost of misstep is imposed with biochemical precision. There is no pursuit, no roar of attack, only the mechanical response of a pressure-triggered syringe embedded within living stone. Within this chemical minefield of the reef, the stonefish represents a different philosophy of toxicity. It does not drift, it does not chase, and it does not broadcast warning colors. It waits. Camouflage forms the first barrier, invisibility shaping the battlefield. Venom stands as the second barrier, activated only when the first fails. Together, They create a layered defense that allows a slow, heavy fish to persist in one of the most competitive ecosystems on Earth. In stillness, it survives. In pressure, it retaliates. At the edge of a coral slope, where blue water darkens into depth, a lionfish drifts forward with unhurried composure, its long pectoral fins fanned wide like an ornate cloak suspended in water. The reef here is alive with movement. Chromis shimmer above branching corals, juvenile wrasses weave through crevices, and parrotfish scrape algae from limestone in steady, rasping rhythms. Most predators in this environment move with caution. They hug the contours of rock, use shadows as cover, and strike only when angles align perfectly. The lionfish, by contrast, advances into open water without hesitation, stripes stark against the reef's mosaic, venomous spines displayed rather than concealed. It behaves not like an ambusher or a sprinter, but like an animal that expects the reef to accommodate it. The dorsal spines rise prominently along its back, each rigid ray connected to venom glands embedded in grooves at their base. Unlike predators whose venom is deployed offensively to immobilize prey, the lionfish's toxin functions primarily as a deterrent. The spines are not concealed or triggered by pressure in the way of a stonefish's defense. They are openly displayed, a visible barricade that communicates risk. The venom induces intense pain and physiological disruption in potential predators, from large reef fish to inquisitive sharks. The cost of biting into a lionfish is high, and many predators appear to learn this quickly. In regions where lionfish are native, few species regularly attempt predation on adults. The spines form a chemical insurance policy that reduces mortality and grants behavioral latitude. This protective chemistry reshapes how the lionfish hunts. Because it faces limited predation risk once mature, it does not need to rush between hiding places or rely exclusively on camouflage. It can patrol slowly over open substrate, inspecting crevices and coral heads at a measured pace. The fins remain extended, not only as a warning but as tools. When approaching a small group of fish near a rock face, the lionfish spreads its large pectoral fins outward, creating a semicircular barrier. This fence behavior compresses the available escape routes for prey, gently herding them toward a vertical surface or into the corner of a coral outcrop. The fish being targeted may attempt to dart sideways, but the visual obstruction and subtle water displacement from the fins guide them into narrower spaces. The lionfish then executes a rapid forward lunge, expanding its mouth in a suction strike that draws the prey inward before it can pivot away. The effectiveness of this hunting strategy relies on calm precision rather than speed. Lionfish do not match the burst acceleration of jacks or barracudas. Instead, they rely on controlled movements that minimize turbulence, allowing them to approach closely without triggering a full panic response in prey. The extended fins also create an illusion of larger body size, which may discourage interference from other predators during feeding. The venomous spines reinforce this impression, transforming what might otherwise be a vulnerable slow hunter into an organism that can operate conspicuously in daylight. This interplay between chemical defense and behavioral boldness becomes especially significant in ecosystems where lionfish have been introduced beyond their native range. In the Western Atlantic and Caribbean, where lionfish are invasive, local predators often lack evolutionary experience with their venomous spines. Juvenile groupers or snappers may approach out of curiosity and suffer painful consequences. Over time, some predators may learn avoidance, but the initial absence of recognition allows lionfish populations to expand rapidly. Their reproductive capacity compounds this advantage. A single female can release buoyant egg masses regularly throughout the year in warm waters, each containing thousands of embryos that drift with currents before settling. High fecundity combined with low predation pressure creates exponential population growth in invaded habitats. The ecological impact extends beyond direct predation. Lionfish consume a wide range of small reef fish and crustaceans, including species that play critical roles in algae control and coral health. By reducing populations of herbivorous fish, lionfish can indirectly influence algal overgrowth on reefs, shifting competitive balance away from coral. The slow, confident hunting style made possible by venom protection allows them to forage extensively during daylight hours when many native predators rest or shelter. Their presence alters prey behavior as well. Small fish may abandon preferred feeding grounds, clustering in suboptimal habitats that offer marginally better refuge from the lionfish's patient approach. The venom itself is protein-based, stored within specialized glandular tissue along each spine. When a predator attempts to bite or swallow the fish, pressure against the spine forces venom along grooves and into the wound. The primary effect is intense localized pain, accompanied by swelling and systemic symptoms depending on exposure. In humans, accidental contact during fishing or diving can result in significant discomfort and temporary impairment. Yet the lionfish does not actively pursue humans, and stings occur almost exclusively through handling or accidental contact. The spines remain a passive deterrent, ready but not deployed unless challenged. Interestingly, juvenile lionfish display more cryptic coloration and may seek shelter more frequently, suggesting that venom alone does not eliminate all risk during early life stages. As individuals grow and spines elongate, confidence increases. Observations in invaded territories often describe lionfish hovering motionless above reef structures, seemingly unconcerned by nearby divers or large fish. This composure is not indifference, but a calculated outcome of chemical security. The cost of testing those spines discourages most potential threats. The relationship between venom and energy expenditure becomes clear when considering metabolic strategy. Active pursuit predators expend significant energy during chases and must compensate with frequent feeding. Lionfish, shielded by venom, can afford a lower intensity hunting rhythm. They conserve energy by moving slowly and exploiting structural features of the reef to corral prey. This approach aligns with their physiology as ectothermic animals whose metabolic rate fluctuates with ambient water temperature. In warmer waters, digestion and activity levels increase, supporting more frequent feeding and reproduction. In cooler conditions, activity slows, but the protective spines remain equally effective, maintaining deterrence regardless of metabolic pace. Predator-prey interactions around lionfish often reveal subtle dynamics. Cleaner fish may approach cautiously, navigating around spines to remove parasites without triggering defensive reactions. This tolerance suggests that lionfish can distinguish between threatening pressure and benign contact, maintaining symbiotic relationships even while heavily armed. The reef ecosystem adapts to their presence, but not always quickly enough in invaded regions. Native species that evolved alongside lionfish recognize the warning silhouette and avoid entanglement. In contrast, naive species may approach too closely, learning through painful encounters or, in some cases, not surviving the lesson. The irony of lionfish success lies in the way chemical defense fosters behavioral audacity. Most reef predators balance risk carefully, using cover and speed to minimize vulnerability. The lionfish, buffered by venom, behaves with visible confidence. It occupies open water columns, hunts during broad daylight, and approaches prey with minimal stealth. This arrogance is not reckless, but enabled by a safety net of toxin that shifts the cost-benefit calculation of exposure. The spines reduce the need for secrecy, allowing a hunting strategy built on patience and spatial manipulation rather than rapid aggression. In regions where lionfish dominate, reef fish communities can shift dramatically. Surveys have documented declines in small native fish densities following lionfish establishment. The absence of natural predators capable of controlling adult lionfish populations amplifies this effect. Human intervention through targeted removal has become a primary management tool in affected areas, reflecting how chemical protection combined with bold foraging can destabilize ecological balance when introduced into new systems. Within the toxic food web, the lionfish represents a distinct model of venom usage. Unlike organisms that manufacture toxins primarily for predatory immobilization, the lionfish uses venom to insulate itself from predation pressure. This insulation frees it to exploit feeding opportunities in ways other species cannot. The reef, normally a landscape of caution and rapid retreats, becomes for the lionfish a domain of measured advance. Its fins extend like banners, its stripes broadcast identity rather than conceal it, and its spines stand as a visible warning that discourages interference. As sunlight filters downward and currents ripple across coral heads, the lionfish continues its deliberate patrol. Small fish scatter and regroup, calculating escape routes that narrow under the spread of those elegant fins. A swift forward surge, a brief vacuum of water, and another prey item disappears. The hunter resumes its poised drift, unhurried and conspicuous, protected by chemistry that transforms vulnerability into dominance. In this corner of the reef, venom does not accelerate the strike. It enables the confidence to strike at leisure. The reef at mid-morning is a lattice of shadows and light, its architecture rising in ridges and shelves where coral heads knit themselves into corridors too narrow for most predators to enter without difficulty. Between these formations, small fish pulse in coordinated bursts, turning as one organism, slipping into crevices the width of a human wrist. It is a landscape designed to frustrate pursuit. A fast predator cannot accelerate freely without colliding with stone. A wide jaw cannot open fully without scraping coral. In this maze, brute force wastes energy. Efficiency wins. From the edge of a coral outcrop, a sea snake glides forward with almost no visible effort. Its body is laterally compressed toward the tail, forming a paddle that slices through water with minimal turbulence. Unlike terrestrial snakes that move by pushing against solid ground, this reptile propels itself through lateral undulations that ripple from head to tail in smooth, measured waves. The motion is deliberate rather than explosive, conserving oxygen stored in an elongated lung that stretches through much of its body cavity. It surfaces occasionally to breathe, but much of its hunting unfolds below in calm, sustained patrols that can last extended periods before another ascent is required. Its head is small relative to body length, the jaws narrow, the gape modest. This is not a constrictor built to wrestle prey into submission. The fish it hunts are sleek, muscular, and perfectly adapted to vanish into crevices the instant they sense disturbance. A prolonged struggle would favor the fish. In the tight geometry of the reef, a snake cannot coil efficiently around a target without entangling itself in coral branches. Evolution has resolved this constraint not by enlarging the snake's jaws or increasing muscular bulk, but by refining a chemical solution. Venom becomes the shortcut. When the snake approaches a cluster of small reef fish sheltering near branching coral, it does not launch into dramatic pursuit. Instead, it threads itself through the coral lattice, body folding and unfolding around obstacles with remarkable spatial awareness. The tongue flickers, sampling chemical traces dissolved in water, while vision guides short, precise adjustments in trajectory. Sea snakes possess specialized sensory adaptations that allow them to detect subtle movements in water pressure, reading disturbances left by passing prey. In a three-dimensional maze, this capacity to interpret hydrodynamic signals can be as valuable as sight. The strike, when it comes, is brief and economical. A forward snap of the head, jaws closing around a fish often no thicker than the snake's own neck. There is no attempt to hold the prey for an extended struggle. Instead, the fangs penetrate quickly, delivering venom deep into muscle tissue before the fish can thrash free. In many species, the venom is primarily neurotoxic and myotoxic, targeting the nervous system and muscle fibers directly. The chemistry disrupts communication between nerve and muscle, interfering with contraction and leading to rapid weakness. The fish may attempt a few reflexive movements, but coordination collapses quickly. The snake maintains a light grip, waiting for physiological failure to do what brute strength cannot. This is not theatrical envenomation. It is calculated efficiency. Venom production requires metabolic investment. Specialized glands synthesize complex proteins and peptides that must be stored and replenished. Although sea snakes possess highly potent venom relative to their size, they do not deploy it indiscriminately. Studies of bite behavior suggest that these snakes can regulate the amount of venom injected, often delivering just enough to incapacitate prey without exhausting reserves. The idea that venomous animals strike recklessly is a misconception rooted in fear rather than observation. In ecological terms, venom is a resource, and like any resource, it must be managed. In the reef's narrow corridors, this efficiency offers clear advantages. A fish that receives a venomous bite and attempts to flee will not travel far before neuromuscular disruption slows its escape. Even if it wedges into a crevice, paralysis may render it incapable of sustained resistance. The snake does not need to chase at high speed, risking injury or energy depletion. It can follow the fading movement, secure in the knowledge that the chemical process is unfolding internally. In this way, venom replaces prolonged wrestling with a timed physiological shutdown. Sea snakes often target burrow-dwelling fish such as gobies or eels that retreat into holes within coral or sand. In these cases, the snake may insert only part of its head into the opening, delivering a rapid bite before withdrawing. The confined space limits physical maneuvering, making constriction impractical. Venom again becomes the decisive factor. A fish immobilized within its own shelter cannot defend itself effectively when the snake returns to retrieve it. The reef's complexity, which shields prey from many predators, becomes less protective when the predator's weapon operates at the cellular level. The energy economics of this strategy are subtle but significant. As air-breathing reptiles, sea snakes must periodically surface, exposing themselves to avian predators and expending energy in vertical travel. A hunting method that minimizes extended chases, conserves oxygen, and reduces time spent at the surface. By delivering venom quickly and allowing its effects to unfold internally, the snake maintains control over the pace of the encounter. The cost of producing venom is offset by the reduced need for sustained muscular exertion. In evolutionary terms, the chemistry has been optimized for the physical constraints of reef hunting. There is also an anatomical trade-off at play. Sea snakes have relatively small heads compared to many terrestrial vipers or cobras, reflecting their specialization on slender fish rather than bulky prey. Large jaws would increase drag and hinder navigation in tight reef spaces. Instead, their skull structure supports precise, rapid bites that place venom exactly where it is most effective. The streamlined head reduces water resistance, enhancing stealth and approach speed. The venom compensates for the modest gape, ensuring that even a brief puncture can neutralize a prey item larger than might otherwise be manageable. The misconception that sea snakes are aggressive or eager to bite humans often arises from their potent venom rather than their behavior. In reality, encounters are rare and typically occur when the snake is handled, stepped on, or trapped in fishing gear. In open water, sea snakes often swim past divers with apparent indifference, their primary focus directed toward prey and navigation. Yet when bites do occur, the physiological consequences can be serious. The same neurotoxic components that incapacitate fish can disrupt human neuromuscular function, leading to muscle weakness, respiratory difficulty, and in severe cases, systemic complications. The danger lies not in frequency of bites, but in the targeted efficiency of the venom's chemistry. Unlike dramatic portrayals of venomous attacks, many sea snake bites are described as initially painless or only mildly painful, which can delay recognition of envenomation. The venom's design prioritizes rapid interference with muscle physiology, rather than causing immediate intense pain. This can create a deceptive window in which symptoms seem minor before systemic effects develop. Again, the venom is tuned for function rather than spectacle. In the wild, dramatic suffering would not increase hunting success. Swift immobilization does. The reef ecosystem responds to the presence of sea snakes in subtle ways. Fish may alter schooling behavior or habitat use when snake density increases, favoring areas with fewer crevices where early detection is possible. Predators of sea snakes, including certain large fish and birds, must contend with the risk of envenomation themselves, influencing predator-prey dynamics higher up the food web. Even the snake's reproduction reflects adaptation to marine life. Many sea snakes give birth to live young in water, bypassing the need to return to land. This continuous aquatic existence allows them to exploit reef systems year-round, maintaining their role as specialized, efficient predators. Observing a sea snake in motion reveals a predator shaped less by aggression and more by constraint. The reef dictates the rules: tight spaces, agile prey, limited oxygen reserves. Venom emerges as the solution that aligns with these rules, allowing a slender reptile to compete successfully in a domain dominated by faster fish. There is a quiet logic to this design. The snake does not overpower the reef, it adapts to its architecture. It does not overpower prey through prolonged struggle. It interrupts physiology at the molecular level. In the broader toxic food web, Sea snakes occupy a position distinct from drifting venomous organisms or toxin-accumulating fish. They manufacture their venom internally, but deploy it with restraint and calculation. Their weapon is not a minefield suspended in currents, nor a passive accumulation of dietary toxins. It is a precisely delivered biochemical tool integrated into a hunting strategy, defined by spatial limitation and energetic balance. The reef remains vibrant and intricate, fish weaving through coral branches, currents carrying plankton overhead. Within this living maze, the sea snake moves as a streamlined silhouette, its venom not a spectacle, but an efficient resolution to the problem of capture in confined space. The water appears harmless at first glance, a pale blue sheet stretched thin over warm coastal sand, sunlight pouring through it without resistance. In these shallows, where swimmers wade and children shuffle their feet along the bottom, the illusion of safety is almost complete. Nothing large patrols here. No shadow moves with predatory intent. Instead, suspended just beyond casual notice, something nearly invisible drifts with the tide. Its body so transparent that it seems less like an animal and more like a distortion in the water itself. This is the box jellyfish, a creature whose danger lies not in size or speed, but in the precision engineering of its venom delivery system. It does not chase with teeth or claws. It manufactures a weapon at the microscopic scale and lets physics carry it into contact. The body is a translucent cube, faintly tinted blue, trailing long, delicate tentacles that can extend many meters behind it. Each tentacle is lined with thousands upon thousands of specialized cells called nematocysts. These are not passive irritants. They are microscopic harpoons, each coiled under internal pressure, each tipped with venom and triggered by the slightest mechanical or chemical signal. When a fish brushes a tentacle, the response is not gradual. It is explosive. Within fractions of a second, thousands of these harpoons fire simultaneously, penetrating skin, embedding in muscle, and injecting venom with astonishing efficiency. It is not the strength of any single sting that matters most. It is the mathematics of contact. The box jellyfish's venom is engineered for speed of effect rather than prolonged suffering. In the dynamic environment of shallow tropical waters, prey cannot be allowed to thrash freely. Currents shift unpredictably. Turbulence from waves can separate predator and prey within moments. A fish that struggles vigorously risks tearing free from fragile tentacles. Evolution has shaped this venom not merely to cause pain or tissue damage, but to interfere rapidly with cellular processes that control muscle contraction and cardiac rhythm. Ion channels within nerve and muscle cells are disrupted, causing immediate dysfunction. Movement slows, coordination collapses, and in many cases, the heart itself is compromised. The fish does not have time to escape the current. It becomes still. This is not brute toxicity, it is strategic incapacitation. In laboratory analysis, the venom contains complex proteins that form pores in cell membranes, altering the electrical balance required for muscle cells to function. The result is rapid paralysis or cardiac distress, depending on dosage and exposure area. In the wild, the effect is visible in seconds. A small fish that brushes multiple tentacles may stop swimming almost instantly, drifting helplessly as the jellyfish's tentacles contract and reel it toward the central mouth. There are no eyes watching in anger, no jaws snapping. The process is mechanical and efficient, more like a trap triggered than a hunt pursued. The concept of contact math becomes clear when observing prey encounters in calm water. A single brush against one tentacle may deliver a survivable dose to a larger fish. But in the chaos of a feeding strike, prey often collides with multiple tentacles at once. Each additional strand represents thousands more injection points. The venom dose multiplies not linearly but exponentially with surface area of contact. The more entangled the prey becomes, the faster physiological systems fail. In shallow bays where currents funnel small schooling fish, the jellyfish does not need to maneuver aggressively. It drifts, tentacles extended like a suspended minefield. The ocean itself increases the probability of contact. Unlike many jellyfish species that drift passively, box jellyfish possess a degree of locomotion unusual among their relatives. Their cube-shaped bell contracts rhythmically, allowing them to swim with directional control. They even possess clusters of simple eyes capable of detecting light and obstacles, enabling them to navigate around mangrove roots or toward darker hunting grounds at dusk. Yet even with this mobility, they remain fundamentally drifting weapons. Tides, wind-driven currents, and thermal gradients do most of the transport work. The energy cost of movement is minimal compared to the return gained when currents concentrate plankton and small fish into narrow corridors. The ocean becomes a conveyor belt that moves the trap for free. This drifting strategy also reduces the need for pursuit-based metabolism. Box jellyfish do not maintain the high-energy physiology of fast predatory fish. Their tissues are largely water, their structure gelatinous and lightweight. The venom does the labor that muscles would otherwise perform. In ecological terms, this is a chemical shortcut. Instead of evolving stronger swimming muscles or more complex jaws, the species invested evolutionary capital into cellular weaponry. The reef and shoreline become zones of invisible hazard, not because the jellyfish dominates physically, but because it weaponizes contact itself. Humans often underestimate the box jellyfish precisely because of this transparency and the familiarity of shallow water. The absence of visible threat leads to relaxed vigilance. A shark silhouette triggers immediate fear. A nearly invisible cube drifting among sunbeams does not. Swimmers step into water waist-deep, unaware that tentacles can extend well beyond the visible body. The first sensation may be a faint brush, followed almost instantly by intense pain, as thousands of nematocysts fire in synchrony. The pattern of injury on human skin often reveals the geometry of the tentacles, long linear welts tracing where contact occurred. The severity of envenomation correlates directly with the length of tentacle exposure. Again, contact math dictates outcome. In severe cases, venom components disrupt cardiac function in humans much as they do in fish. Rapid cardiovascular collapse has been documented in extreme exposures, particularly when large surface areas are stung. Yet many incidents occur in calm, shallow beaches, reinforcing the illusion that danger resides only in deeper offshore waters. The ecology of the box jellyfish contradicts this assumption. They often inhabit nearshore environments because prey density is high, and because mangrove-lined coasts provide shelter from heavy surf. These same environments are attractive to humans seeking warm, clear water. The overlap is ecological coincidence rather than aggression, but the result can be catastrophic when contact area is high. Beyond individual encounters, the box jellyfish plays a subtle but important role in regulating small fish populations along tropical coastlines. By selectively removing weaker or slower individuals that collide with tentacles, they contribute to predation pressure that shapes schooling behavior and spatial distribution of juvenile fish. In areas where jellyfish populations surge, fish may alter migratory timing or avoid certain inshore habitats altogether. This behavioral ripple affects larger predators that depend on those fish, subtly influencing food web dynamics. A drifting, gelatinous predator can shift patterns of energy flow in ways disproportionate to its apparent fragility. There is also a seasonal rhythm to their presence. In many regions, box jellyfish populations increase during warmer months when reproductive cycles align with optimal plankton availability. Warmer water temperatures can accelerate metabolic processes, supporting faster growth of juveniles. As coastal nutrient levels fluctuate with rainfall and river input, prey abundance changes, indirectly influencing jellyfish distribution. Their life cycle includes a benthic polyp stage attached to substrate, capable of asexual reproduction before releasing free-swimming medusae. This dual lifestyle anchors them briefly to the seafloor before releasing them again into the drifting phase. The transformation from stationary polyp to mobile venomous medusa is a dramatic shift from passive existence to active chemical weapon. Observing a box jellyfish in open water is an exercise in restraint. It does not lunge. It does not display aggression. It pulses gently, tentacles trailing with deceptive grace. The danger is statistical rather than theatrical. A fish that never touches a tentacle passes unharmed. A fish that grazes one lightly may survive. But a fish that blunders into multiple strands becomes an equation with only one likely result. The ocean's movement ensures that eventually something will intersect the suspended web of stinging cells. In the broader context of the toxic food web, the box jellyfish represents pure manufacture. It synthesizes its venom internally, packaging it into disposable delivery units that can fire once and then regenerate. Each nematocyst is a single-use syringe, but there are so many that loss is inconsequential. The body continuously replaces expended cells, maintaining a full arsenal at minimal energetic cost. Unlike animals that accumulate toxins from diet, this species invests directly in production, controlling both dose and deployment through mechanical sensitivity. It is less a passive drifter than a floating battery of microscopic projectiles. The reef at its edge becomes a chemical minefield, not through visible markers, but through probability fields, where currents slow, where fish schools gather, where swimmers wade without suspicion. The invisible cube may drift. Its presence transforms open water into contested territory governed by contact geometry and cellular chemistry. In this system, strength is irrelevant, speed secondary, and visibility almost non-existent. The weapon is preloaded, the trigger hair-thin, and the ocean itself carries it patiently through the shallows, waiting for the mathematics of touch to complete the equation. The lagoon floor appears calm beneath the filtered sunlight, its pale sand rippled by gentle currents that erase footprints as quickly as they form. Patches of seagrass sway in slow, synchronized motion, and small invertebrates burrow and resurface in a rhythm dictated by tides. Nothing in this shallow expanse suggests urgency or threat. Yet just beneath a thin veil of sand, a living disk lies nearly indistinguishable from the substrate itself. The outline is subtle, a faint asymmetry in the ripple pattern. Two slightly raised spiracles, pulsing rhythmically to draw water over concealed gills. The stingray does not advertise its presence. Its first defense is invisibility. The body of the ray is dorsoventrally flattened. Pectoral fins expanded into broad wings that merge seamlessly with the head. This shape allows it to settle into sediment with minimal profile, covering itself in sand through gentle undulations that lift and resettle grains across its back. Only the eyes and spiracles remain exposed, positioned atop the body so that respiration continues, while the mouth and gill openings remain pressed against the substrate. Camouflage here is functional and dynamic. The ray adjusts its burial depth according to light intensity, predator presence, and temperature, balancing concealment with the need to monitor its surroundings. Most interactions between stingrays and larger animals unfold without incident. Rays are primarily benthic foragers, feeding on mollusks, crustaceans, and small fish that dwell within the sediment. Electroreceptors embedded in their skin detect faint bioelectric signals generated by muscle contractions of hidden prey. When a clam shifts or a shrimp flexes beneath the sand, the ray perceives the disturbance and pivots slightly, using its flexible disc to trap prey before crushing it with robust dental plates. The mouth is located ventrally, reinforcing the orientation toward the seafloor rather than toward open confrontation. Yet the same posture that facilitates feeding also creates vulnerability. A ray lying buried is difficult to detect from above. Predatory sharks, large fish, and even wading mammals may pass within close proximity. The ray's solution is not pursuit or warning display, but a final layer of deterrence mounted along its tail. This structure, commonly referred to as a barb, is a modified spine composed of calcified tissue with sharp serrations along its edges. It is not a biting mechanism. It does not lunge forward. Instead, it is positioned for a rapid upward or backward flick when the animal perceives imminent threat from above. The distinction between mechanical injury and venom injection is central to understanding the stingray's defensive strategy. The spine is encased in a thin integumentary sheath that contains venom-producing tissue. When the spine penetrates flesh under force, the sheath ruptures, allowing venom to enter the wound. The serrations along the spine can tear tissue as it enters, and in some cases, as it withdraws. This combination of laceration and toxin distinguishes stingray injuries from simple puncture wounds. The physical damage creates a pathway for venom to spread, while the venom compounds the trauma by inducing pain and inflammation. Stings most commonly occur not during active pursuit, but through surprise contact. In shallow coastal waters, humans wading barefoot may inadvertently step near or directly on a buried ray. The downward pressure from a foot compresses the ray's body and restricts escape routes. In response, the ray reflexively whips its tail upward. The motion is swift and controlled, designed to dislodge the threat rather than to pursue it. If contact is made, the spine can puncture deeply, driven by the force of the step itself. Much like the stonefish's pressure-dependent spines, the severity of injury correlates with applied force. Body weight drives the barb further into tissue, increasing both mechanical damage and venom delivery. The venom of stingrays is protein-based and thermolabile, meaning it can be denatured by heat. This characteristic underlies common first aid practices involving hot water immersion, which may alleviate pain by disrupting toxin structure. Biologically, the venom induces intense localized pain, likely serving as an immediate deterrent to predators. In fish or sharks, such pain would discourage continued attack. In humans, the pain can be severe and disproportionate to the apparent size of the wound. Swelling and tissue damage follow, reflecting both the mechanical tearing from serrations and the inflammatory response triggered by venom components. Despite their formidable defense, stingrays are not aggressive animals. Observational studies reveal that rays often flee when given sufficient warning. When approached slowly by divers, many rays lift from the substrate and glide away in a fluid, undulating motion. The problem arises when the warning window is eliminated. A sudden step near a buried ray leaves little time for escape. The tail strike is not a targeted attack, but a defensive reflex shaped by evolutionary pressures in environments where ambush predators are common. The ecological context of this defense becomes clearer when considering predator interactions. Large sharks are among the few natural predators capable of consuming stingrays. Some shark species exhibit specialized behaviors to avoid the spine, flipping rays over to access the unprotected underside before biting. This maneuver demonstrates the arms race dynamic between predator and prey. The ray's venomous barb deters casual predation, but determined predators evolve strategies to circumvent it. In this balance, venom functions as a selective filter, discouraging inexperienced or opportunistic attacks while failing against highly adapted specialists. Camouflage remains the primary line of defense. By blending into the sediment, rays reduce the likelihood of detection altogether. The venomous spine becomes relevant only when camouflage fails. This layered defense system mirrors patterns observed in other reef organisms, where invisibility precedes chemical deterrence. The ray's body coloration often matches the surrounding sand or seagrass beds, and some species can adjust pigment distribution slightly to enhance blending. The ability to remain motionless for extended periods conserves energy and reduces visual cues that might attract predators. Thermoregulation plays a subtle role in stingray behavior in shallow lagoons. As ectothermic animals, rays absorb ambient heat from warm sand and water, influencing metabolic rate and digestive efficiency. In cooler conditions, activity levels decline, and rays may remain buried longer to conserve energy. During warmer periods, increased metabolism supports more frequent foraging. Seasonal movements between shallow nursery areas and deeper waters reflect reproductive cycles and temperature preferences. Many species use shallow bays as birthing grounds, where juveniles benefit from reduced predator presence and abundant benthic prey. Juvenile rays possess proportionally smaller spines and may rely more heavily on camouflage and rapid escape than on envenomation. As individuals mature, the spine enlarges and venom gland development increases defensive capability. In some species, the spine may be shed and replaced periodically, maintaining sharpness and functionality. This regenerative aspect ensures that the mechanical component of defense remains effective despite wear or breakage. The combination of injury and venom carries ecological implications beyond immediate deterrence. Predators that survive encounters with stingrays may develop avoidance behaviors, altering hunting routes and prey selection patterns. Such learned avoidance can shape spatial distribution of both rays and their predators, influencing benthic community structure. In heavily trafficked coastal areas, human awareness campaigns encourage shuffling feet while wading to alert buried rays and reduce surprise encounters. This simple behavioral adjustment reflects an understanding of the ray's reliance on surprise as the trigger for defensive action. Within the broader toxic food web, stingrays represent a fusion of mechanical and chemical defense rather than a purely venom-driven strategy. The barb itself is an anatomical adaptation capable of causing significant trauma. The venom enhances this trauma, ensuring that even partial penetration delivers a memorable deterrent. Unlike predators that use venom to subdue prey rapidly, the ray's toxin reinforces a boundary. It communicates through pain and injury that contact is costly. In the tranquil shallows where sunlight dances across sand ripples, the stingray remains mostly unseen. A slight depression in the substrate, a pair of protruding eyes, and a tail lying flat behind the body are the only indicators of life beneath the surface. Most of the time, interactions proceed without consequence. The ray feeds, the tide shifts, and the ecosystem hums with quiet continuity. Only when weight descends unexpectedly does the defensive machinery engage, converting a peaceful lagoon into a moment of sharp lesson. The real danger, then, lies not solely in venom or in the spine, but in their integration. Mechanical force drives penetration. Serrations tear tissue. Venom floods the wound. Each component amplifies the other, forming a composite defense shaped by evolutionary pressures in crowded coastal ecosystems. Camouflage conceals. Venom punishes. Between these layers, the stingray persists as a largely misunderstood inhabitant of shallow seas, neither hunter of humans nor passive ornament, but a benthic specialist whose survival depends on remaining unseen until contact makes deterrence unavoidable. The reef at dusk carries a different tension than it does at noon. Colors soften, shadows lengthen, and the architecture of coral becomes a maze of negative space rather than vibrant sculpture. Within crevices and across limestone ledges, dark, rounded forms begin to emerge, extending fine, articulated spines outward in every direction. Where the reef once appeared smooth, it now bristles. Sea urchins, previously wedged deep into cracks to avoid daylight predators, creep outward to graze. The substrate transforms into a field of moving needles. At first glance, a sea urchin seems simple, a globe encased in spines. Yet that globe is a rigid shell known as a test, composed of interlocking calcium carbonate plates arranged with mathematical precision. Each spine articulates on a ball and socket joint, capable of subtle adjustments in angle and orientation. When threatened, an urchin does not lunge or chase. Instead, it pivots its spines toward the source of disturbance, creating a defensive hemisphere that presents only sharp points to the intruder. In some species, between the long primary spines are shorter, specialized structures called pedicellariae, tiny pincer-like appendages that can grasp and, in certain species, deliver venom through microscopic jaws. The effect is not dramatic in the way of a stingray's strike or a jellyfish's discharge. It is incremental, persistent, and strategically placed. The power of sea urchin defense lies less in a single spine and more in collective geometry. A predator attempting to bite down encounters not a smooth surface, but a barrier of rigid spikes that distribute force across multiple contact points. Each spine can break off and remain embedded, causing irritation and potential infection. In venomous species, these embedded spines introduce compounds that intensify pain or inflammation. The message is clear. A quick, opportunistic snap at a sea urchin becomes costly. Even if the predator is not seriously injured, the discomfort and risk of mouth damage discourage repetition. This cost reshapes predator behavior across the reef. Fish that might otherwise graze freely over certain substrates alter their routes when urchin densities rise. Groupers and wrasses skirt around heavily populated areas, focusing instead on open patches where prey can be pursued without negotiating a forest of spines. The reef floor becomes partitioned not only by coral structures, but by defensive organisms that modify movement patterns. In areas dense with urchins, the battlefield changes. Narrow corridors between coral heads are effectively lined with deterrents. A predator lunging after a small fish risks colliding with spines, making what might have been an effortless capture into a hazardous maneuver. For smaller predators, the calculation is even more restrictive. A triggerfish, equipped with strong jaws and a willingness to experiment, may attempt to flip a sea urchin onto its dorsal side. The underside lacks the same density of long spines, and once inverted, the urchin struggles to right itself. This flipping technique represents a behavioral innovation, a learned method to bypass the primary layer of defense. In temperate regions, sea otters demonstrate even more sophisticated strategies, diving to the sea floor to collect urchins and then floating on their backs while cracking the tests open with stones balanced on their chests. Tool use transforms an armored grazer into a manageable meal. These predator adaptations illustrate the dynamic interplay between mechanical defense and cognitive or anatomical countermeasures. Despite such strategies, the presence of abundant urchins alters ecological balance significantly. Sea urchins are voracious grazers, feeding primarily on algae that grow on rock and coral surfaces. Using a specialized feeding structure called Aristotle's lantern, a complex arrangement of 5 calcareous teeth, they scrape surfaces with remarkable efficiency. In moderate numbers, urchins contribute to reef health by controlling algal overgrowth, preventing algae from smothering corals. However, when predator populations decline or environmental conditions favor urchin reproduction, population booms can occur. In these scenarios, grazing pressure intensifies dramatically. An urchin boom can convert lush, algae-rich reefs into barren landscapes known as urchin barrens. The substrate becomes stripped of macroalgae, leaving behind bare rock and encrusting coralline algae. While this may initially seem beneficial for coral recruitment, excessive grazing can also inhibit recovery by removing juvenile coral recruits or destabilizing the balance of benthic organisms. The reef's visual character changes, and so does its functionality. Fish species that depend on algal cover for food or shelter may decline. In this way, the defensive success of urchins cascades into structural and trophic shifts across the ecosystem. The defensive strategy of sea urchins also includes behavioral elements beyond spine orientation. Many species display negative phototaxis, avoiding bright light and retreating deeper into crevices during the day. This movement reduces exposure to visually hunting predators. At night, when many reef fish reduce activity, urchins venture farther from shelter to graze. Their tube feet, small hydraulic appendages extending from the test, grip the substrate and coordinate slow, deliberate movement. The pace is measured, almost imperceptible over short intervals, yet over the course of a night, an urchin may traverse significant distances in search of food. Some urchin species engage in a behavior known as covering, in which they pick up fragments of shell, algae, or debris and place them atop their bodies using tube feet and spines. This covering may serve multiple functions: shading from ultraviolet radiation, camouflage from predators, or stabilization against currents. The act of covering transforms the urchin into a mobile extension of the substrate, further blurring the distinction between organism and environment. This blending reinforces the concept that the reef itself becomes spiked as the defensive organism merges visually and physically with its surroundings. Venomous components in certain tropical species add another layer of deterrence. Pedicellariae equipped with toxin glands can snap shut on contacting tissue, delivering localized venom that causes pain and inflammation. Although rarely life-threatening to humans, these structures amplify the defensive envelope of the urchin. A careless hand brushing against what appears to be inert substrate may encounter both penetrating spines and pinching venomous appendages. The interaction is seldom intentional from the urchin's perspective. It is a reflexive response shaped by millions of years of predation pressure. The thermoregulation of sea urchins reflects their ectothermic physiology and benthic lifestyle. Because they lack the ability to regulate internal temperature actively, their metabolic rate aligns closely with ambient water conditions. In warmer waters, feeding rates increase, accelerating grazing impact. In cooler environments, activity slows, but spines remain as effective deterrents. Seasonal changes in temperature and nutrient availability can influence reproductive cycles, leading to pulses of larval release into the plankton. These larvae drift with currents before settling onto suitable substrate, potentially colonizing new reef areas. The dispersal stage connects distant ecosystems, spreading defensive architecture across broad geographic ranges. Predator populations exert significant control over urchin numbers. In regions where key predators such as large fish or sea otters decline due to overfishing or habitat loss, urchin populations may expand unchecked. This expansion exemplifies how removal of top-down pressure can allow a well-defended grazer to reshape entire habitats. The transformation is not subtle. Once established, dense aggregations of urchins create a physical and behavioral barrier that discourages predator entry, reinforcing their dominance. Observing a reef section densely populated by sea urchins conveys a palpable shift in atmosphere. Fish navigate cautiously, adjusting swimming trajectories to avoid accidental contact. A grazing parrotfish hesitates before descending into a patch thick with spines. The structural complexity of coral remains, but the accessibility changes. The battlefield has been modified not by aggression, but by deterrence. The reef floor, once open terrain for pursuit and grazing, now demands careful choreography. Within the broader toxic food web, sea urchins represent a form of environmental fortification. Their defenses are primarily mechanical, augmented in some species by venomous elements, and distributed across space rather than concentrated in a single organism's movement. Unlike predators that carry toxin as a weapon for targeted encounters, urchins anchor their defense to the substrate, altering the cost landscape for every other participant. The presence of spines increases the price of careless foraging, making cheap attacks expensive. As night deepens and currents whisper across the reef, the urchins continue their slow advance, scraping surfaces clean and reshaping microhabitats. Predators adapt, either learning to flip, bite carefully at the unprotected underside, or in some cases employing tools to breach the armored sphere. The arms race persists quietly beneath the surface. Yet for much of the reef's inhabitants, the message is constant. This ground is defended. Spines radiate outward, not in pursuit, but in declaration. In their collective presence, the reef becomes a field of deterrence, a living terrain where chemistry and structure converge to redefine movement, access, and survival. At first light, the reef seems motionless, a cathedral of coral pillars and overhangs suspended in clear water. Yet along the rock faces and within sheltered depressions, clusters of soft, tentacled forms sway with deliberate rhythm, their columns anchored firmly to limestone, while their crowns bloom outward into the current. A sea anemone is neither plant nor coral, though at a distance it resembles both. Its body is a muscular tube crowned by a ring of tentacles, each lined with microscopic stinging cells that wait for contact. In the gentle surge of water, those tentacles flex and curl, not searching, not pursuing, but holding position in a flow that does the hunting for them. The anemone's strategy is built upon stillness. It cannot chase prey across open sand, nor can it lunge with the speed of a fish. Instead, it establishes territory in locations where water movement is predictable. Reef crests, tidal channels, and lagoon mouths provide consistent currents that carry plankton, small fish, and drifting invertebrates past its waiting arms. The animal positions itself where delivery is reliable, converting hydrodynamics into opportunity. The ocean becomes conveyor belt and accomplice, transporting potential meals directly into reach. Each tentacle is lined with nematocysts, specialized stinging cells shared across the cnidarian lineage. When triggered by mechanical or chemical cues, these cells discharge microscopic harpoons loaded with toxin. The initial contact is swift and nearly invisible, but the effect on small prey is immediate. The toxin disrupts cellular membranes and interferes with neuromuscular coordination, stunning or paralyzing the victim. Yet unlike free-swimming jellyfish, the anemone adds a second layer to its capture system. The tentacles are not merely stinging filaments, they are adhesive and muscular, capable of curling around prey and holding it long enough for the venom to take full effect. This combination of chemical and mechanical capture defines the anemone as a living glue trap. A small fish that brushes against one tentacle may attempt to dart away, only to encounter adjacent tentacles that cling and sting simultaneously. The more it struggles, the more contact points activate. The adhesive mucus secreted by the tentacles increases friction, preventing escape even before paralysis sets in. The prey's own thrashing spreads it deeper into the ring of tentacles, drawing it closer to the central mouth located at the top of the column. The mouth then opens, and through coordinated contraction of the body wall, the anemone transfers the immobilized prey inward for digestion. The digestion process unfolds slowly within the gastrovascular cavity, where enzymes break down tissue externally before absorption occurs. Because the anemone relies on intermittent delivery rather than constant pursuit, its metabolism is adapted to variable feeding intervals. Extended periods without prey do not necessarily threaten survival. Energy reserves accumulate when currents are rich with plankton, and digestion proceeds methodically. In this way, the anemone exemplifies an energy-efficient predator, investing in stationary infrastructure rather than high-speed musculature. The reef responds to the presence of anemones with distinct behavioral adjustments. Fish that are vulnerable to stings often maintain a cautious distance, learning to recognize the tentacled crown as a hazardous boundary. In densely populated reef zones, anemones can create localized no-go areas, pockets of substrate avoided by grazing fish and wandering invertebrates. These zones alter traffic patterns, influencing where small fish congregate and where predators choose to hunt. A predator pursuing prey near an anemone must calculate the risk of accidental contact, and many choose instead to redirect pursuit into safer corridors. This spatial influence becomes more complex when considering the symbiotic relationship between certain fish species and sea anemones. Clownfish, for example, inhabit the tentacles of specific anemone species, protected from stings by a specialized mucus coating that inhibits nematocyst discharge. This partnership represents a rare negotiation within the reef's chemical politics. The fish gains refuge from predators that would otherwise consume it, while the anemone benefits from nutrients in fish waste and from the fish's movements that enhance water circulation around tentacles. The presence of clownfish can also lure curious predators closer, increasing the likelihood of prey contact with tentacles. What appears at first to be an unlikely friendship reveals itself as a strategic alliance shaped by chemical compatibility. The immunity of clownfish is not innate at birth. Juveniles must acclimate gradually to the host anemone, brushing tentacles gently to acquire protective mucus and avoid triggering full envenomation. This process underscores the specificity of chemical communication at the microscopic level. The anemone distinguishes between prey and partner based on surface chemistry and mechanical cues, maintaining defensive readiness while tolerating selected inhabitants. In doing so, it reinforces its territorial presence, creating microhabitats within its tentacle field. Environmental factors such as temperature and water chemistry influence anemone physiology significantly. As ectothermic organisms, anemones experience metabolic shifts in response to ambient temperature changes. Warmer waters accelerate enzymatic reactions, potentially increasing feeding rates when prey is abundant. However, prolonged thermal stress can disrupt symbiotic relationships with photosynthetic algae that reside within some species' tissues. These algae contribute nutrients through photosynthesis, supplementing the anemone's diet. When water temperatures rise beyond tolerance thresholds, bleaching can occur, reducing energy input and weakening the organism. Thus, even a stationary predator is intricately linked to broader climatic conditions. Reproductive strategies further illustrate adaptation to variable environments. Many sea anemones reproduce both sexually, releasing gametes into the water column for external fertilization, and asexually through budding or fission. A single individual can divide, producing genetically identical clones that colonize adjacent substrate. Over time, this process can create dense clusters of anemones, amplifying their spatial influence. A reef ledge that once hosted a single tentacled crown may transform into a contiguous field of stinging polyps, reinforcing the no-go zone effect. Predators capable of consuming anemones must navigate both mechanical stings and chemical defenses. Certain nudibranches and butterfly fish species feed on anemones, often exhibiting resistance to nematocysts. These predators have evolved physiological adaptations that allow them to ingest or avoid toxins without severe consequence. In some cases, Nudibranchs even incorporate undischarged nematocysts into their own tissues, repurposing the stinging cells for defense. This recycling of chemical weaponry highlights the fluidity of toxin rolls within the reef's food web. The anemone's sensory world operates without centralized vision or hearing. Instead, it relies on a diffuse nerve net capable of detecting touch and chemical gradients. This decentralized system allows rapid local responses to tentacle contact while maintaining overall structural integrity. When disturbed by strong currents or mechanical agitation, the anemone can retract tentacles and contract its column, reducing exposure. Such responses are energy efficient and reversible, enabling survival in environments subject to tidal surges and occasional storm-driven turbulence. In regions where nutrient levels fluctuate seasonally, anemones adjust feeding patterns accordingly. During plankton-rich periods, tentacles may extend fully and remain active for extended durations. In leaner times, contraction reduces metabolic demand. This flexibility supports persistence across diverse reef conditions. Although anchored to a single point, the anemone participates dynamically in ecosystem flux. Within the broader toxic food web, the sea anemone represents a form of passive aggression structured around patience and chemistry. It does not roam the reef in search of conflict. Instead, it establishes a zone of influence and waits. The currents deliver opportunity, and the tentacles enforce boundaries. Chemical deterrence shapes fish movement, predator pursuit, and even social alliances through symbiosis. As daylight fades and plankton rise in the water column, the tentacles extend once more, glowing softly under the last rays of sun. Tiny organisms drift closer, unaware of the adhesive field awaiting them. A small fish strays too near, contact is made, and the sequence unfolds without spectacle, but with certainty. The living glue trap closes gently, methodically, drawing nourishment from a system powered by flow rather than chase. In this quiet architecture of the reef, the anemone stands as both hunter and boundary, a fixed point around which the choreography of avoidance and alliance revolves. On a reef slope where sunlight fractures into rippling patterns across coral heads, a single sea anemone anchors itself in a pocket of rock, its tentacles extended into the passing current like a living thicket. To most fish, that thicket is a boundary not to be crossed. The tentacles pulse gently, each one armed with microscopic stinging cells ready to fire at the slightest provocation. Small wrasses veer away at the last moment, damselfish circle at a respectful distance, and even larger predators adjust their path to avoid accidental contact. Within this chemical perimeter, movement should mean pain, paralysis, and ingestion. Yet from deep within the tentacles, a flash of orange emerges, weaving confidently between the stinging filaments as if they were soft blades of grass rather than lethal weapons. The clownfish does not appear built for dominance. Its body is compact, its fins rounded, its swimming style more flutter than glide. It lacks venomous spines, crushing jaws, or the sleek acceleration of open water predators. And yet, within this 1 square meter of reef, it behaves as if the surrounding space belongs entirely to it. The secret lies not in toxicity, but in compatibility. The clownfish survives inside a living trap, not by resisting venom in the traditional sense, but by altering the chemical conversation that triggers it. Sea anemones fire their stinging cells when they detect a combination of mechanical disturbance and specific chemical cues on a surface that signal prey. The outer layer of a typical fish carries mucus containing compounds that anemone tentacles recognize as foreign and edible. Contact initiates a cascade. Nematocysts discharge, venom injects, tentacles contract. The clownfish disrupts this sequence at the earliest step. Its skin is coated in a specialized mucus layer that lacks or masks the chemical triggers that would normally provoke discharge. In some species, the mucus may even incorporate compounds derived from the host anemone, effectively cloaking the fish in the anemone's own chemical identity. This compatibility is not automatic at birth. Juvenile clownfish approaching an anemone for the first time do so with deliberate caution. They brush tentacles lightly, retreating, repeating the contact gradually to build tolerance. During this acclimation process, the fish modifies its mucus coating, integrating the host's chemical signature. Once established, the immunity is functional rather than absolute. Violent, prolonged disturbance can still provoke stings, but routine movement within the tentacles proceeds without incident. The anemone perceives the fish not as prey, but as background. Living inside a weapon transforms the clownfish's ecological reality. Predators that might easily swallow a small orange fish hesitate at the edge of the tentacle field. A snapper lunging for the clownfish risks brushing multiple stinging filaments, triggering pain and confusion. The clownfish exploits this hesitation. It darts outward to feed on plankton and small invertebrates, rarely venturing far from the tentacle perimeter. When threatened, it retreats instantly into the anemone's embrace, weaving deeper into the living barrier. The home base is not a cave or a crevice, it is an active chemical defense system that punishes intrusion. This arrangement alters survival probabilities dramatically. In open reef habitat, a fish of similar size without protection would face constant predation pressure. Mortality rates for small reef fish are typically high, especially during juvenile stages. The clownfish's partnership reduces that risk substantially, enabling longer lifespans and stable breeding territories. The anemone benefits as well. The fish's movements increase water circulation through the tentacles, enhancing oxygen exchange and removing debris. Waste products from the fish provide nutrients, contributing to the host's metabolic needs. In some cases, clownfish may even lure larger fish toward the anemone, increasing opportunities for accidental prey capture. Territorial behavior in clownfish is shaped by this secure base. Within the tentacle field, the fish exhibits aggression disproportionate to its size. It charges at intruding fish many times larger than itself, nipping and pursuing them beyond the immediate boundary before retreating. This boldness is not reckless, it is calculated. The clownfish knows that any pursuer must confront the anemone's defenses to continue the chase. The weaponized habitat amplifies the fish's confidence, allowing it to defend a small but resource-rich patch of reef effectively. Social structure among clownfish adds further complexity. Groups typically consist of a dominant breeding pair and several smaller subordinate individuals. The largest fish is female, the next largest male, and the remainder are non-breeding juveniles. Should the female die, the dominant male undergoes sex change, becoming female and assuming reproductive leadership. This sequential hermaphroditism ensures continuity within the limited territory of the anemone. Because suitable host anemones are finite resources, competition for occupancy is intense. Territorial defense is not merely about feeding rights, but about access to reproductive space. The interplay between size hierarchy and territorial aggression is tightly regulated. Subordinates maintain smaller body size through behavioral suppression and limited food access, reducing conflict within the group. The anemone becomes a structured community center, its tentacles sheltering not only individuals, but social dynamics shaped by chemical refuge. Predators observing from a distance may see only flickers of orange within a forest of tentacles, unaware that a complex hierarchy operates beneath that canopy. Thermoregulation in clownfish follows the patterns of other reef fish, with metabolic rates rising in warmer waters. Because anemones often inhabit sunlit shallows, temperature fluctuations can influence both partners. Elevated water temperatures may stress the anemone, especially if it hosts symbiotic algae sensitive to thermal extremes. Bleaching events can weaken the host, indirectly affecting the clownfish's security. In such cases, the fish may relocate to alternative anemones if available, though competition for unoccupied hosts can be fierce. Migration across open reef exposes the fish to heightened predation risk, underscoring how deeply survival is tied to chemical compatibility with a specific host. Predator interactions around clownfish reveal subtle patterns. Some species have learned to approach anemones cautiously, attempting to snatch clownfish during brief excursions beyond the tentacles. Success depends on timing and precision. The clownfish counters with rapid retreat and aggressive feints. The presence of venomous tentacles complicates every attack, creating a spatial puzzle for would-be predators. Even if a predator manages to seize a clownfish, escape through the tentacle field may still be possible if the predator cannot follow safely. The immunity of clownfish does not extend to all anemone species universally. Chemical compatibility is often species-specific, reflecting coevolution over extended timescales. In regions where clownfish are absent, anemones function solely as predatory organisms without resident fish. In areas where multiple clownfish species coexist, each may associate with particular anemone hosts, reducing direct competition. These associations illustrate how chemical signaling shapes community composition across the reef. Within the broader toxic food web, the clownfish represents a departure from the pattern of manufacturing or accumulating toxins. It does neither. Instead, it leverages another organism's chemical arsenal by blending into it. Immunity becomes its superpower, not through internal resistance to venom, but through avoidance of triggering the weapon at all. The distinction is subtle but profound. The clownfish does not neutralize toxin after exposure. It prevents exposure from occurring through surface chemistry. As currents continue to sweep across the reef, the anemone's tentacles remain extended, indifferent to the small fish weaving among them. Plankton drifts past, some captured, others passing through. A larger fish approaches and halts at the perimeter, reconsidering the cost of entry. Within this living fortress, the clownfish patrols its territory with energetic defiance, chasing intruders and returning to the safety of its stinging canopy. Its survival is not a product of size or speed, but of chemical negotiation. In a reef where toxins define boundaries and shape behavior, the clownfish stands as an example of strategic compatibility. It has not evolved sharper teeth or thicker armor. It has evolved the ability to belong inside a weapon that others fear. The tentacles that immobilize prey become protective walls. The venom that deters predators becomes an extension of its own defense. In this arrangement, chemistry does not merely protect, it empowers, transforming a small, brightly colored fish into the confident guardian of a toxic sanctuary. At the edge of a reef wall where shadow deepens into indigo, an octopus emerges from a narrow crack in the limestone. Its body changes color as it moves, mottled browns blending into speckled gray, texture shifting from smooth to granular, as though it were testing different identities against the stone. Nothing about its shape is fixed. The mantle contracts and expands gently as it ventilates water across its gills, while 8 arms unfurl in deliberate arcs, each one exploring the substrate ahead. There are no fangs on display, no obvious weapons protruding from its body. Yet, concealed beneath the flexible skin, lies a beak and a delivery system of venom that operates with precision rather than spectacle. Unlike predators that seize prey with teeth or crush with powerful jaws, the octopus approaches hunting as a coordinated exercise in control. It does not chase fast-moving fish across open water. Instead, it navigates crevices, probing cavities where crabs wedge themselves and bivalves anchor tightly to rock. The arms function as both sensory instruments and restraints. Along their inner surfaces, rows of suction cups detect chemical traces and subtle movements. Each sucker is capable of independent adjustment, gripping, tasting, and manipulating with remarkable dexterity. When an arm slips into a hole and encounters resistance, the octopus does not pull blindly. It assesses. The venomous bite is delivered through a chitinous beak located at the center of the arm cluster. Once prey is secured within the arms, the octopus positions the beak with surgical accuracy. In many species, venom contains neurotoxins capable of disrupting neural transmission in prey, inducing paralysis or at least profound weakness. The bite is not intended to tear large chunks of flesh. It is a targeted injection designed to reduce struggle. In the case of a crab, the octopus may pierce the softer membrane at the base of a limb or between segments of the exoskeleton, exploiting anatomical vulnerabilities rather than brute force. The venom travels quickly, interfering with motor control, allowing the octopus to maintain its grip without enduring prolonged resistance. This reduction of struggle is not merely convenient, it is protective. Crabs and lobsters possess formidable claws capable of inflicting serious injury. An octopus has no rigid skeleton to shield it, only pliable tissue and a soft mantle. A powerful snap from a claw could tear skin or damage an eye. By weakening prey rapidly, the octopus minimizes the risk of counterattack. The venom functions as risk management, transforming potentially dangerous encounters into manageable tasks. Once the prey is subdued, The beak can work methodically, cracking shell or slicing tissue while the arms hold the body steady. Shell-bearing prey present a different challenge. Clams and mussels seal themselves tightly when threatened, relying on strong adductor muscles to resist opening. An octopus confronted with a closed bivalve may apply multiple strategies. It can insert the tip of a single arm into the narrowest gap, applying persistent pulling force. It may also use its radula, a rasping tongue-like structure, to drill a small hole through the shell, injecting venom directly into the interior cavity. The drilling behavior demonstrates both patience and anatomical awareness. The octopus positions itself to maintain leverage while minimizing exposure to potential predators, often working under the cover of a rock ledge. Venom in this context reduces the energetic cost of prying open resistant prey. Once injected, the toxin relaxes the bivalve's muscles, allowing the shell to gape. The octopus then inserts its beak and consumes the soft tissue within. This approach illustrates how venom complements mechanical manipulation. It is not a dramatic external display, but an internal sabotage of muscle control. The octopus does not overpower the shell. It persuades it to fail. Predator-prey interactions on the reef reveal the octopus as both hunter and hunted. Groupers and moray eels may search crevices for octopuses, attracted by movement or scent. To survive, the octopus employs camouflage that rivals any reef inhabitant. Chromatophores within its skin expand or contract to match surrounding patterns almost instantly, while papillae alter skin texture to mimic rock, coral, or algae. This capacity to disappear reduces encounters before they escalate. Yet if confronted directly, the octopus may release ink, creating a cloud that confuses the predator's vision and scent detection, allowing escape. The reliance on venom, rather than powerful jaws, aligns with the octopus's overall physiology. As a cephalopod, it possesses a decentralized nervous system with a significant portion of neurons located within the arms themselves. Each arm can execute complex movements semi-independently, enabling coordinated restraint of prey. The beak remains centrally located, but its effectiveness depends on the arm's ability to position and secure targets. Venom enhances this choreography by ensuring that once contact is made, the prey's capacity to retaliate diminishes quickly. The problem-solving dimension of octopus hunting extends beyond physical manipulation. Observations in both laboratory and wild settings reveal that octopuses can learn from repeated encounters. They may remember the location of particularly productive hunting grounds or the structure of specific crevices that house prey. In tidal pools, individuals have been seen stacking stones around their den entrances, creating barricades against intrusion. When hunting within complex reef matrices, an octopus may block alternative escape routes for a crab before committing to the final strike, placing one or more arms strategically to seal exits. This anticipatory behavior suggests an awareness of prey movement patterns and a capacity to plan sequences of action. Venom also plays a role in rare but significant interactions with vertebrate prey. Some species target small fish, enveloping them within a web of arms before delivering a bite. The neurotoxic components can immobilize fish rapidly, preventing thrashing that might alert larger predators. In certain blue-ringed octopus species, venom potency extends to vertebrates, including humans, containing tetrodotoxin that blocks sodium channels in nerve cells, leading to paralysis. Although most octopus species are not dangerously venomous to humans, the existence of such potent chemistry within the group underscores the versatility of venom as a tool. Thermoregulation in octopuses reflects their ectothermic nature and reliance on ambient water temperature. Metabolic rates increase in warmer waters, potentially enhancing hunting frequency and digestion speed. However, elevated temperatures can also increase vulnerability to hypoxia as oxygen solubility decreases. Octopuses respond behaviorally, seeking shaded crevices or deeper water to moderate exposure. Because their blood uses hemocyanin rather than hemoglobin to transport oxygen, they are particularly sensitive to temperature-induced changes in oxygen availability. This physiological nuance influences where and when they hunt. Reproduction introduces additional ecological dynamics. Many octopus species have relatively short lifespans, with females dedicating substantial energy to brooding eggs. During this period, a female may guard a clutch within her den, foregoing feeding and relying on stored reserves. The use of venom during hunting prior to reproduction supports energy accumulation needed for this terminal investment. Once eggs hatch, juveniles disperse into the plankton or settle into benthic habitats, beginning independent lives without parental guidance. Within the broader toxic food web, the octopus stands apart as a predator that wields venom discreetly and intelligently. It does not advertise toxicity through warning colors, nor does it rely on venom as a territorial deterrent. Instead, it integrates chemical incapacitation into a broader strategy of manipulation and restraint. The reef's complexity, with its labyrinthine cracks and armored prey, demands adaptability rather than speed alone. The octopus answers this demand with cognitive flexibility and biochemical assistance. As the tide shifts and currents whisper along the reef wall, the octopus retreats momentarily into its den, arms folding inward as though reassembling into a single, breathing stone. Shell fragments scattered near the entrance testify to previous hunts, evidence of strategic breaches rather than brute force collisions. When it ventures out again, the skin shifts color to match new terrain, and the arms resume their exploratory dance. Somewhere within the next crevice, a crab braces itself against intrusion, unaware that precision rather than power is approaching. Venom in this context is neither spectacle nor excess. It is a calculated component of a hunting system designed to reduce risk and conserve energy. By weakening prey at the neural level, the octopus protects its vulnerable body while enabling intricate manipulation. It is a reminder that in the reef's chemical economy, intelligence and toxin often work hand in hand, quietly reshaping the balance between predator and prey. In a shallow reef lagoon, where coral branches interlace like frozen lightning and soft corals sway with tidal rhythm, a creature no longer than a human thumb moves with unhurried precision across the substrate. Its body glows in saturated blues and incandescent oranges, edged in electric lines of white that seem almost artificial against the muted stone. It appears fragile, even ornamental, as though it were an accidental flourish of color in an otherwise functional ecosystem. Yet this sea slug is neither decorative nor defenseless. It is a specialist in theft, a quiet tactician that builds its arsenal not by manufacturing toxins, but by appropriating the weapons of others. The sea slug belongs to a group of nudibranchs, known for their elaborate forms and vivid pigmentation. Their external anatomy is soft and exposed, lacking shell or rigid armor. Along their backs rise clusters of finger-like projections called serrata, extensions of the digestive system that serve multiple purposes. These serrata increase surface area for respiration, house digestive glands, and in certain species function as repositories for something far more unusual: stolen stinging cells. The story begins with prey that most reef animals avoid. Hydrozoans and other small cnidarians cling to coral rubble and seagrass blades, their delicate tentacles armed with nematocysts capable of firing venom-tipped harpoons at microscopic invaders. To a small fish, these colonies represent minor hazard. To a human, perhaps an irritating brush. To the sea slug, they are opportunity. It approaches the stinging colony deliberately, rasping tissue away with its radula, a ribbon-like structure lined with minute teeth. As it consumes the cladarian flesh, something remarkable occurs within its digestive tract. Instead of digesting every component of its prey, the slug selectively transports undischarged nematocysts through specialized channels toward the tips of its serrata. These stinging cells remain structurally intact and functional. The slug integrates them into sacs called cnidosacs, where they are stored without triggering discharge. This phenomenon, known as kleptocnidae, transforms the sea slug into a living vault of borrowed ammunition. The nematocysts remain viable and ready to fire, now positioned not on the original owner's tentacles, but on the slug's own protruding extensions. The physiological feat required for this process is profound. The slug must prevent nematocysts from discharging prematurely within its own tissues. It achieves this through controlled transport mechanisms and chemical compatibility that inhibit activation. The same cellular triggers that would cause discharge upon contact with foreign surfaces are somehow suppressed during ingestion and relocation. Once housed in the serrata, however, the nematocysts are once again capable of firing when stimulated by a predator's touch. This appropriation of weaponry allows the sea slug to become dangerous without synthesizing venom itself. It invests energy not in toxin production, but in careful harvesting and maintenance of a chemical arsenal sourced entirely from prey. The elegance of this strategy lies in efficiency. Rather than evolving a complex venom gland system, the slug leverages an existing one, co-opting the defensive technology of its food source. It becomes, in effect, a mobile extension of the cnidarian colony it consumes. The serrata serve as both deterrent and display. When threatened, the slug can arch its body and orient the serrata toward the aggressor, increasing the likelihood of contact with stinging tips. Some species can autotomize serrata, detaching them if grasped, leaving behind a cluster of firing nematocysts while the slug escapes. The detached serrata continue to discharge stings for a short period, reinforcing the idea that even fragments of this animal carry consequence. Coloration in these sea slugs often functions as aposematic signaling, warning potential predators of chemical defense. The brilliant hues are not camouflage in the conventional sense, yet within the chaotic palette of a reef, bright coloration can paradoxically blend into the visual noise created by sponges, corals, and encrusting algae. What appears conspicuous to a human eye may fragment into disruptive patterns underwater. For predators that have learned through painful experience, however, the colors broadcast a clear message. Contact will not be without cost. Predator-prey dynamics surrounding these slugs illustrate the success of stolen defenses. Fish that might sample other soft-bodied invertebrates often avoid vividly colored nudibranches altogether. In experimental observations, naive predators may attempt to bite, only to recoil rapidly after encountering firing nematocysts. Over time, learned avoidance reduces attack frequency. The slug's vulnerability as a shell-less organism is mitigated by a chemical shield that does not originate from within. The ecological implications extend beyond individual survival. By preying on cnidarian colonies, sea slugs regulate the growth and distribution of these stinging organisms. In areas where nudibranch populations rise, hydrozoan coverage may decline, subtly altering microhabitats for other invertebrates. The slug's theft thus feeds back into reef structure, balancing the abundance of organisms that themselves influence coral competition and space occupation. Thermoregulation and metabolic considerations shape the slug's activity patterns. As ectotherms, sea slugs adjust feeding rates according to ambient water temperature. Warmer conditions may accelerate digestion and nematocyst transport, enhancing defensive capacity. However, elevated temperatures can also stress both slugs and their cnidarian prey, influencing availability of functional stinging cells. Seasonal shifts in prey abundance may determine where certain slug species congregate, leading to localized concentrations of both predator and borrowed weaponry. The sensory world of a sea slug is subtle yet effective. Rhinophores, paired sensory organs extending from the head, detect chemical gradients in the water, guiding the slug toward prey colonies. Vision is limited, but chemical cues provide detailed information about the surrounding environment. The slug navigates slowly, following invisible trails that lead to stinging prey others avoid. In doing so, it converts hazard into resource. Some species exhibit remarkable specificity, feeding exclusively on particular cnidarian hosts. This specialization ensures compatibility between ingested nematocysts and storage mechanisms. The evolutionary partnership is intimate. The slug's survival depends on access to prey that provide functional stings, and the prey's distribution influences slug range. Migration patterns for these slugs are often limited to short distances within reef systems, tracking the growth of their cnidarian hosts. Larval dispersal may extend range, but successful settlement requires proximity to appropriate prey colonies. The irony embedded in this system is subtle yet profound. The sea slug, lacking shell or overt physical defense, appears vulnerable in nearly every anatomical respect. Yet through chemical appropriation, it achieves a level of deterrence comparable to inherently venomous organisms. It becomes dangerous without manufacturing danger. The energy cost saved by avoiding toxin synthesis may be redirected toward growth, reproduction, or further specialization. Reproductive strategies among nudibranchs add another layer of intrigue. Many are simultaneous hermaphrodites, possessing both male and female reproductive organs. During mating, individuals exchange sperm reciprocally, maximizing reproductive opportunity in sparse populations. Egg masses are often laid in coiled ribbons attached to substrate near food sources, ensuring that hatchlings emerge into environments rich with cnidarian prey. The continuity of stolen defense thus depends on the continuity of feeding behavior across generations. Within the broader toxic food web, the sea slug stands as a master of second-hand power. It does not drift as a weapon, nor does it deploy venom in a predatory strike. It harvests and redeploys. The reef becomes a marketplace of chemical assets, where stinging cells change hands and shift allegiance. A cnidarian colony that once deterred predators on its own now indirectly shields a soft-bodied grazer that feeds upon it. As currents sweep across the reef and sunlight fades into deeper hues, the slug continues its deliberate progress over coral rubble. Its serratas sway gently, each tip housing microscopic harpoons ready to fire. A curious fish approaches and pauses, perhaps recognizing the warning colors, perhaps recalling a previous encounter. It turns away. The slug advances unchallenged, armored not by shell or speed, but by borrowed chemistry integrated seamlessly into its flesh. In this final tableau of the toxic food web, the sea slug embodies an unexpected perspective. Danger need not be self-produced to be effective. Sometimes survival lies in understanding another organism's weapon deeply enough to make it one's own. The reef, intricate and layered, thrives on such exchanges, where boundaries between predator and prey blur, and stolen tools redefine what it means to be defended. On the quiet margin between coral heads and open sand, where the reef loosens its grip and gives way to sediment, a sea cucumber lies stretched along the seafloor like a discarded length of mottled rope. Its body is soft, cylindrical, and unassuming, colored in muted browns and greens that mirror the substrate. Small tube feet ripple along its underside, gripping grains of sand as it inches forward in slow, deliberate motion. There is no armor plating, no visible spines, no flashing warning colors. In a landscape shaped by teeth, venom, and rapid escape, the sea cucumber appears almost recklessly vulnerable. Yet this apparent defenselessness conceals a strategy built not on striking back, but on making attack fundamentally unappealing. The sea cucumber is an echinoderm, kin to sea stars and sea urchins, but its body plan has elongated into a flexible cylinder. Beneath the pliable skin lies a framework of tiny calcareous ossicles embedded within connective tissue, providing subtle structural support without rigidity. The mouth faces forward along the substrate, encircled by tentacles that gather organic detritus and microscopic life from the sand. Feeding is methodical. The cucumber ingests sediment, extracts nutritional particles through a digestive tract that winds through its length, and expels processed sand behind it. In doing so, it functions as a recycler of the reef, aerating sediment and redistributing nutrients across the benthic environment. This slow procession across the seabed exposes the sea cucumber to constant risk. It cannot flee swiftly from a prowling fish. It cannot bite or sting. Its thermoregulation follows the rhythm of the water, as an ectotherm whose metabolism rises and falls with temperature. In warmer currents, feeding accelerates. In cooler conditions, movement slows further. The vulnerability is real. Yet predators that experiment with sampling a sea cucumber often abandon the effort abruptly. The reason lies not in visible weaponry, but in chemistry released directly into the water. Many sea cucumber species produce compounds known broadly as saponins, surface-active molecules that interact disruptively with cell membranes. These substances are not delivered through fangs or spines. Instead, they are embedded within the body wall and can be released into surrounding water when the animal is stressed or physically disturbed. When a fish attempts to bite into the soft body, these compounds leach outward, creating a localized chemical cloud. The immediate effect on the predator is irritation. Gill tissues, highly sensitive and permeable, encounter the dissolved toxins first. Discomfort and physiological disruption follow, prompting the fish to retreat. The mechanism operates less like a venomous injection and more like a smoke grenade deployed underwater. The sea cucumber does not need to wound its attacker directly. It simply changes the chemical composition of the water in its immediate vicinity. The predator's own respiratory system becomes the point of contact, amplifying exposure as water flows continuously across the gills. In concentrated form, these compounds can impair membrane function, destabilizing cells and discouraging continued aggression. The predator learns quickly that biting into this slow-moving organism carries consequences disproportionate to the nutritional reward. In some species, defensive escalation involves the expulsion of specialized internal structures known as Cuvierian tubules. When severely threatened, the sea cucumber contracts its body and releases these sticky filaments through the cloaca, projecting them outward into the water column. The tubules elongate rapidly upon contact with seawater, forming a tangled mass that adheres to potential attackers. This behavior is not an act of retaliation, but of distraction and deterrence. The predator, confronted with adhesive threads and chemical irritation, must pause to clear its mouth or gills, providing the cucumber time to contract or wedge itself into crevices. The regeneration capacity of sea cucumbers complements this defensive strategy. Loss of Cuvierian tubules or partial body tissue is not necessarily fatal. Over time, the animal can regenerate expelled structures, restoring its chemical arsenal. This regenerative ability reflects an evolutionary trade-off. Instead of investing energy in armor or speed, the sea cucumber invests in resilience and deterrence. If attacked, it can afford to sacrifice parts of itself temporarily, confident in its capacity to recover. Predator behavior around sea cucumbers illustrates the success of this approach. Many reef fish species that graze actively across the substrate ignore sea cucumbers entirely, focusing instead on more palatable invertebrates. Even opportunistic predators that sample a cucumber once often avoid repeating the experience. This learned avoidance reduces predation pressure across the population. The sea cucumber becomes, in ecological terms, a poor decision. It is not physically formidable, but it is chemically inconvenient. The presence of sea cucumbers influences sediment ecology in subtle but significant ways. By ingesting and excreting sand continuously, they aerate the substrate, preventing compaction and promoting microbial activity. This bioturbation enhances nutrient cycling, supporting benthic microfauna and indirectly benefiting adjacent coral communities. In areas where sea cucumber populations decline due to overharvesting, sediment quality can deteriorate, leading to shifts in microbial composition and reduced resilience of the reef system. Thus, the survival of this slow-moving detritivore carries broader ecological weight. Thermoregulation again intersects with defense. Elevated water temperatures can increase metabolic stress, potentially altering toxin production and release rates. Some studies suggest that environmental stresses influence the concentration of saponins within body tissues. This dynamic relationship between environment and chemical defense underscores the sensitivity of sea cucumbers to climate shifts. Because they lack rapid escape behaviors, their survival hinges on maintaining effective deterrent chemistry, even as conditions fluctuate. Reproductive strategies among sea cucumbers often involve broadcast spawning, where males and females release gametes into the water column in synchrony. In some species, individuals raise the anterior portion of their body into the current during spawning events, increasing dispersal efficiency. This brief elevation, exposing more of the body above the substrate, momentarily increases vulnerability. Yet the same chemical defenses that deter predators during routine feeding remain active, providing protection even during reproductive displays. The balance between exposure and deterrence remains intact. Rarely, certain fish species have evolved tolerance to sea cucumber toxins, feeding selectively on specific species without severe consequence. These interactions highlight the ongoing arms race within the reef. A chemical defense effective against most predators may be circumvented by specialists capable of detoxification or selective feeding techniques. Nevertheless, the generalist predator community continues to treat sea cucumbers with caution. The visual subtlety of sea cucumbers belies the complexity of their internal chemistry. Their skin, often textured and covered in papillae, serves as both physical interface and chemical reservoir. When disturbed, the release of saponins into surrounding water creates a transient boundary that shifts the immediate environment. Unlike venom that requires direct contact, these compounds disperse outward, influencing not only the attacking predator, but any organism in close proximity. The defense radiates rather than pierces. In the broader toxic food web, the sea cucumber represents a different philosophy of survival. It does not chase, sting, or bite. It does not advertise danger through brilliant coloration. Instead, it relies on chemical deterrence that alters the decision-making calculus of potential predators. The reef floor becomes a place where not all prey are worth pursuing. A fish scanning the substrate for edible invertebrates weighs options, and the sea cucumber often falls low on the list. As twilight deepens and nocturnal feeders emerge, the sea cucumber continues its slow passage across the sand, processing sediment grain by grain. A curious fish approaches, nips tentatively, then withdraws abruptly, flicking its fins in agitation. The cucumber contracts slightly, perhaps releasing a subtle chemical pulse, then resumes its steady advance. There is no dramatic confrontation, no visible clash of predator and prey. There is only the quiet assertion of chemistry in water. To survive as a slow animal in a fast world requires redefining the terms of engagement. The sea cucumber accomplishes this not by overpowering threats, but by convincing them to reconsider. In doing so, it maintains its role as both recycler and resident within the reef's intricate tapestry. The battlefield shifts not through violence, but through deterrence, and the soft-bodied crawler persists by becoming the choice few predators are willing to make twice. Across a warm tropical reef flat where seagrass blades ripple in the tide and coral bommies rise like scattered fortresses, a pufferfish moves with deliberate calm. Its body appears almost comical in proportion, rounded and compact, patterned in subtle mosaics of tan, olive, and cream that break up its outline against sand and coral. It does not swim with the streamlined efficiency of a wrasse or the muscular thrust of a jack. Instead, it propels itself using small, rapid oscillations of its pectoral fins, maneuvering with precision rather than speed. Its eyes rotate independently, scanning above and behind as it grazes on small crustaceans, mollusks, and fragments of algae. At first glance, it seems like one of the reef's more awkward inhabitants, slow and exposed in a world where hesitation can be fatal. Yet the pufferfish embodies a layered defense strategy that merges mechanical transformation with biochemical consequence. The first line of defense is visible and immediate. When startled, the puffer draws water into its highly elastic stomach, expanding its body into a near spherical shape. The transformation is rapid, powered by strong muscles that control intake and retention of water. The fish's spines, normally flattened against the skin in many species, extend outward as the body inflates, turning the once modest form into a bristling orb. This sudden enlargement alters the predator's calculation. A mouth sized to engulf a compact fish now confronts an object too large or too awkward to swallow. The inflation alone can be sufficient to deter some predators. A grouper lunging at a puffer may find its jaws unable to close around the expanded body, forcing a retreat. But the puffer's defense does not end with mechanical exaggeration. Beneath the skin and within certain organs resides tetrodotoxin, a potent neurotoxin that interferes with the fundamental process of nerve signaling. The toxin does not cause dramatic tissue destruction or visible injury. Instead, it targets sodium channels embedded in nerve cell membranes, preventing the propagation of electrical impulses required for muscle contraction. In sufficient quantities, this disruption leads to paralysis. What makes the pufferfish particularly intriguing within the toxic food web is that tetrodotoxin is not always synthesized by the fish itself. Research suggests that the toxin is often associated with symbiotic or environmental bacteria acquired through diet. Marine bacteria capable of producing tetrodotoxin inhabit sediments and certain invertebrates. As puffers consume organisms such as small crustaceans and mollusks that harbor these bacteria, the toxin accumulates within their tissues. Over time, the fish concentrates the compound in specific organs, including the liver, skin, and in some species, the ovaries. The puffer becomes toxic not solely by internal manufacture, but by integration within a microbial and dietary network. This microbial linkage reframes the puffer's toxicity as part of a broader ecological circuit. The reef is not merely a stage of predator and prey interactions, it is also a chemical marketplace where bacteria contribute to the defensive arsenals of higher organisms. The puffer's immune system and physiology must tolerate and compartmentalize the toxin without succumbing to its effects. Specialized binding proteins within the fish's tissues may neutralize tetrodotoxin internally, preventing it from interfering with the puffer's own nerve conduction. This internal resistance underscores the evolutionary negotiation between host and toxin source. The combination of inflation and tetrodotoxin creates a defense stack that discourages experimentation. A predator that attempts to bite an inflated puffer may ingest toxin in the process. Even sublethal exposure can induce numbness or weakness, reinforcing avoidance learning. In ecosystems where puffers are common, many predators appear to recognize the distinctive body shape and avoid pursuit altogether. The fish's leisurely movements reflect this chemical confidence. It does not need to dart constantly between hiding places. Instead, it patrols open areas, relying on its layered defenses to offset its lack of speed. The ecological consequences extend further. Because puffers feed on hard-shelled invertebrates using robust beak-like teeth formed by fused dental plates, they influence populations of crabs, snails, and other benthic organisms. Their foraging activity helps regulate species that might otherwise proliferate and overgraze algae or compete with coral recruits. In this way, a fish protected by toxin participates actively in maintaining reef balance. The toxin does not transform it into a passive ornament. It supports an ecological role that might otherwise be untenable given its vulnerability. Thermoregulation plays a subtle role in toxin dynamics. As ectotherms, puffers experience metabolic changes in response to water temperature. Warmer conditions may increase feeding rates, potentially influencing the accumulation of tetrodotoxin if toxin-bearing prey are abundant. Conversely, environmental stresses that alter microbial communities in sediments could affect toxin availability within the food chain. This interdependence highlights the fragility of chemical defenses linked to ecological networks. The puffer's toxicity is not isolated. It reflects microbial health and dietary pathways within its habitat. Human interaction with pufferfish reveals the dual nature of fascination and risk. In certain cultures, specially trained chefs prepare pufferfish as a delicacy, carefully removing organs known to contain high concentrations of tetrodotoxin. Improper preparation can leave residual toxin in edible portions, leading to paralysis in consumers. Symptoms typically begin with tingling sensations and may progress to muscular weakness and respiratory compromise if significant quantities are ingested. The danger does not stem from an aggressive act by the fish, but from mishandling of its chemical defenses. The toxin's potency lies in its capacity to silence nerve communication without causing overt tissue damage, making early symptoms deceptively mild before more severe effects manifest. Predator-prey interactions around puffers often involve hesitation rather than confrontation. Sharks and large fish may approach, circle, and then veer away upon recognizing the distinctive shape. In some instances, inexperienced predators may attempt to bite and quickly release the fish, retreating after encountering spines and possible toxin exposure. The puffer's inflation response is energetically costly and cannot be sustained indefinitely, but it need only last long enough to discourage the immediate threat. Once the predator withdraws, the fish expels the ingested water and resumes its rounded form, the spines flattening once more against its skin. Interestingly, some species of pufferfish utilize tetrodotoxin not only for defense, but potentially for reproductive signaling. Studies have suggested that trace amounts of the toxin may function as chemical cues during mating, influencing aggregation behavior. This possibility reflects the multifaceted role of toxins beyond simple deterrence. Compounds that deter predators may also mediate communication within species, illustrating the complex chemical dialogues that permeate marine ecosystems. Migration patterns among puffers vary by species. Some remain localized within reef territories, while others undertake seasonal movements to spawning grounds. During such migrations, exposure to unfamiliar predator communities may test the effectiveness of their defense stack. Yet the universal deterrent properties of tetrodotoxin and inflation often transcend local predator familiarity, offering protection across habitats. Within the broader toxic food web, the pufferfish embodies an intersection of microbial partnership, mechanical adaptation, and chemical deterrence. It does not drift passively like a jellyfish, nor borrow stinging cells as a sea slug does. Instead, it accumulates toxin through diet and harbors it within its own tissues, integrating microbial chemistry into vertebrate physiology. The reef around it becomes a space where caution prevails, where predators recognize the silhouette of a fish that is more trouble than it is worth. As currents shift and twilight descends across the lagoon, the puffer continues its unhurried patrol, nibbling at encrusting organisms and probing the sand for hidden prey. A larger fish glides nearby, assessing, then turning away. The puffer remains composed, its layered defenses quietly reinforcing its presence. In a world defined by speed and aggression, it survives by transforming itself into a biochemical paradox. Slow, yet secure. Vulnerable in form, yet formidable in consequence. In the blue clarity just beyond the reef's jagged skyline, a barracuda hangs suspended like a polished blade, left floating in mid-water. Its body is long and rigid, silver flanks reflecting the sun in hard flashes as it turns slightly to keep the reef in view. The jaw is angular, lined with interlocking teeth built for seizing rather than chewing. Nothing about its appearance suggests toxicity. It is streamlined, clean, a predator defined by speed and precision rather than by spines or warning colors. Smaller fish scatter when it advances, not because it carries visible chemical weapons, but because it carries intent. The barracuda is an apex ambush hunter in many reef systems. It patrols the edge between open water and coral structure, waiting for schools of smaller fish to drift into striking range. When it moves, it does so in sudden acceleration, body stiffening into a controlled surge that can outpace most prey within a short burst. The strike is efficient. The fish is seized, often swallowed whole or torn quickly into manageable portions. In this immediate interaction, there is nothing toxic about the barracuda. It does not inject venom or sting. It relies on muscle, hydrodynamics, and timing. Yet beneath that sleek exterior lies a possibility shaped not by its own biology, but by the chemistry of the reef. The toxin associated with barracuda is not produced by the fish itself. It originates at the smallest scales of the ecosystem, in microscopic dinoflagellates that grow on algae covering coral and rocks. These single-celled organisms produce compounds that can interfere with sodium channels in nerve cells, disrupting the flow of electrical signals. The toxin moves upward through the food chain in increments so small they are invisible to the eye, yet significant in cumulative effect. The process begins when herbivorous fish graze on algae that host toxin-producing dinoflagellates. The dinoflagellates themselves are minute, clinging to surfaces and thriving in warm, nutrient-influenced waters. A small grazing fish consumes thousands of these cells over time, accumulating low levels of toxin within its tissues. That fish may show no outward sign of contamination. It swims normally, feeds, and avoids predators like any other reef inhabitant. But the toxin does not degrade easily. It persists within muscle and organs, slowly concentrating. When a larger predatory fish consumes many of these smaller fish, the toxin load increases further. This process, known as biomagnification, amplifies chemical presence at each trophic level. By the time a top predator, such as a large barracuda, has fed repeatedly across seasons and years, the accumulated concentration can become significant. The barracuda remains unaffected behaviorally. Its physiology tolerates the toxin without paralysis. It continues to hunt, patrol, and reproduce as if nothing unusual resides within its tissues. From the outside, the fish appears pristine. Its scales gleam, its movements are confident, its eyes track prey with focused intensity. There is no visual cue that it carries a chemical legacy of the reef's microbial community. Yet to a human who catches and consumes such a fish, the effect can be profound. Ciguatera poisoning arises not from venom injected by the fish, but from ingestion of flesh that has concentrated toxin through diet. The compound interferes with nerve signaling, leading to a range of symptoms that can include gastrointestinal distress and neurological disturbances. In severe cases, it alters sensory perception in ways that can be disorienting and prolonged. What makes this phenomenon particularly compelling within the toxic food web is that the barracuda itself is not an intentional participant in chemical warfare. It is a carrier, a vessel shaped by ecological promotion of toxins upward through feeding relationships. The reef's smallest producers, invisible without magnification, influence the safety of apex predators. Environmental factors such as water temperature, nutrient runoff, and reef disturbance can influence dinoflagellate growth. Following storms or coral bleaching events, shifts in algal communities may create favorable conditions for toxin-producing species. Thus, the barracuda's chemical burden reflects not only its diet, but the broader health and balance of the reef. Predator-prey interactions remain unchanged in appearance. A school of sardines flickers near the reef edge. The barracuda lunges, teeth closing around a flash of silver. The captured fish is swallowed. In that act, another increment of toxin may be added to the predator's tissues. Over time, these increments accumulate. The process is invisible, silent, and cumulative rather than immediate. The barracuda does not deploy toxin as defense or offense. It becomes dangerous to humans by promotion through trophic levels. The concept of biomagnification extends beyond barracuda to other large reef predators such as groupers and snappers. However, barracuda often gain particular notoriety due to their size and position near the top of the reef's food web. Larger individuals, having fed for many years, are more likely to carry higher toxin loads. This relationship between size and chemical accumulation adds a paradox to fishing practices. The most impressive specimen, admired for its length and weight, may also carry the highest risk. From a physiological perspective, the resilience of barracuda to these toxins suggests evolutionary tolerance. Their sodium channels may differ subtly in structure from those of mammals, reducing binding efficiency of the toxin. Alternatively, they may sequester the compounds in tissues that limit neurological impact. Regardless of mechanism, the fish remains functionally unaffected in its own environment, continuing to fulfill its ecological role as predator. Thermoregulation and migration patterns influence exposure risk. In warmer tropical waters, where dinoflagellate populations thrive, the likelihood of toxin presence increases. Barracuda that migrate between reefs may experience variable exposure depending on local algal communities. Juvenile barracuda, feeding on smaller prey closer to shore, may accumulate toxin gradually as they grow and shift toward larger prey. The interplay between diet breadth and toxin accumulation illustrates how feeding ecology shapes chemical identity over time. To a diver observing a barracuda in the water column, none of this chemical narrative is visible. The fish appears as a pure embodiment of predatory design. Elongated body, forked tail, minimal drag. It often hovers motionless, using subtle fin adjustments to maintain position in current. The reef below bustles with herbivores grazing and invertebrates filtering water. The barracuda watches, waiting for the right moment to strike. It is a predator defined by clarity and efficiency, not by overt toxicity. And yet in certain contexts, it carries a hidden burden. The toxin within its tissues serves no purpose in its own survival. It is not a deterrent to sharks or a tool for subduing prey. It is a byproduct of its position at the top of a chemically dynamic ecosystem. In this sense, the barracuda becomes an emblem of how interconnected the reef truly is. The health of microscopic algae can reverberate upward to influence human experience far beyond the water's edge. Within the toxic food web, this chapter reveals a different form of danger. Unlike the deliberate manufacture of venom by a jellyfish, or the strategic theft of stinging cells by a sea slug, the barracuda's toxicity is incidental. It is promoted by diet, accumulated through trophic transfer, and stored without intent. The fish itself remains unaware of its chemical cargo, moving through blue water as it always has. As the sun descends and the reef shifts toward evening calm, the barracuda drifts near the surface, its silhouette sharp against fading light. Small fish gather in tighter schools, wary of sudden acceleration. The predator's body reflects the last glints of gold before darkness deepens. It is immaculate in form, Yet within its flesh resides the echo of countless microscopic organisms consumed indirectly through the chain of life. The barracuda does not sting or inject. It simply carries the reef's chemistry upward, transforming invisible toxins into a hidden consequence of ecological promotion. Along the shadowed lip of a reef wall, where coral shelves overhang like eroded balconies and the blue water beyond deepens into cobalt, a massive grouper rests almost motionless. Its body is thick and muscular, patterned in mottled browns and pale speckles that echo the texture of surrounding limestone. At first glance, it appears less like a hunter and more like a patient fixture of the reef itself, a quiet bulk wedged between coral pillars. Yet beneath that still exterior is a predator engineered for sudden force, capable of turning an entire cave mouth into a trap. The grouper's strategy is rooted in proximity and patience. It does not chase prey across open water like a barracuda, nor does it rely on venomous deterrents. Instead, it inhabits structural complexity, caves, crevices, and overhangs, where small fish seek shelter from currents and larger predators. The reef's architecture becomes its advantage. Hovering just inside a shadowed cavity, the grouper aligns its body with the entrance, eyes scanning the shifting silhouettes of passing fish. Its fins make minute adjustments to hold position, conserving energy in a posture that appears relaxed, but remains coiled with readiness. When a target drifts too close, the grouper's mouth expands with explosive precision. The jaws open wide, and the buccal cavity enlarges rapidly, generating a powerful suction force that pulls water and prey inward. This feeding method, known as suction feeding, allows the grouper to engulf fish before they can react. The strike is brief, almost silent, and the prey vanishes into the cavernous mouth. The predator then closes its jaws and returns to stillness. Digestion beginning in darkness, within a body that rarely advertises its dominance. In this behavior lies the foundation for an unexpected chemical narrative. The grouper is not venomous. It does not sting or inject toxin. Yet in certain reef systems, it can become a carrier of ciguatera toxins, accumulating them through diet over years of feeding. The process begins at the base of the reef's productivity, among microscopic dinoflagellates that colonize algae growing on coral rubble. These microorganisms produce compounds capable of interfering with sodium channels in nerve cells, disrupting the normal flow of electrical impulses. Herbivorous fish grazing on algae ingest these dinoflagellates, incorporating small amounts of toxin into their tissues. For a small grazing fish, the toxin concentration may remain low enough to cause no visible impairment. The fish swims, feeds, and evades predators as usual. But when a grouper consumes that fish, it acquires not only muscle and energy, but also a trace of the accumulated toxin. Over time, as the grouper feeds repeatedly on herbivores and smaller carnivores that have themselves eaten grazers, the toxin concentration within its tissues increases. The process is gradual and cumulative, a chemical ascent through trophic levels. Groupers are particularly suited to becoming reservoirs of this accumulation because of their feeding habits and life history. Many species grow slowly and can reach substantial size over decades. A large individual may have consumed thousands upon thousands of smaller fish across its lifespan. Each meal contributes incrementally to the toxin load. Time becomes a variable in chemical risk. The longer a fish lives and the higher it feeds within the food web, the greater the potential for biomagnification. The reef's physical structure amplifies this effect. Groupers often establish territories centered around caves that function as feeding hubs. Schools of smaller fish seek shelter within these same structures, especially during tidal shifts or predator presence. The grouper's ambush method ensures efficient capture of prey concentrated by habitat. In essence, the cave becomes both pantry and trap, allowing the predator to collect toxin-bearing fish with minimal energy expenditure. The efficiency of suction feeding means that prey are swallowed whole, including organs where toxins may be stored, further increasing accumulation. Unlike organisms that actively use toxins for defense or predation, the grouper's chemical burden is incidental. It does not derive immediate advantage from carrying ciguatoxins. The toxin does not deter sharks or prevent competition from other predators. Instead, it represents an ecological byproduct of its position as a reef boss, an apex ambusher integrating the chemical history of the community into its own flesh. From the outside, there is no sign of this accumulation. The grouper's scales are intact, its movements powerful and deliberate. It commands respect within the reef, not because of visible hazard, but because of size and territorial dominance. Smaller fish give it wide berth. Even divers approaching cautiously can feel the weight of its presence as it hovers at the mouth of its domain. Its mouth, large enough to engulf surprisingly substantial prey, remains slightly open as water passes over gills in steady rhythm. Thermoregulation and environmental conditions influence the broader dynamics of toxin production. Warmer waters can favor the proliferation of dinoflagellates, especially following disturbances such as coral bleaching or storm damage that increase available algal surfaces. In such contexts, the base level of toxin entering the food chain may rise. Groupers feeding in these regions may accumulate higher concentrations over time. Migration patterns can also play a role. Some grouper species undertake seasonal movements to spawning aggregation sites, traveling across reef systems with varying toxin prevalence. Individuals may therefore experience differing levels of exposure depending on their feeding grounds. Human interaction with large reef fish often reflects cultural and economic values. A substantial grouper caught by hook or spear represents both food and achievement. Yet in regions known for ciguatera prevalence, consuming large reef predators carries risk. The toxin is heat-stable, meaning cooking does not neutralize it. Symptoms in humans can include gastrointestinal distress and neurological effects, such as altered temperature sensation, where cold feels hot, and vice versa. These experiences arise from the toxin's interference with sodium channels in nerve cells, echoing the same molecular target that defines its ecological impact. The paradox lies in the fish's appearance. A large grouper looks robust and wholesome, a symbol of reef abundance. Its flesh appears clean and firm. There is no visible warning coloration, no spines or inflation display. The danger, when present, is invisible and silent, embedded within muscle fibers accumulated over years. In this sense, the grouper mirrors the reef itself, beautiful and productive on the surface, yet chemically complex beneath. Predator interactions within the reef rarely revolve around toxin in the case of groupers. Sharks may prey on smaller individuals, and larger species may compete for territory. The grouper's defense is primarily size and the security of its cave. Juveniles, lacking the mass of adults, often occupy shallower or more protected habitats, reducing predation risk while they grow. As they mature and claim larger territories, their feeding range expands, and with it the diversity of prey consumed. Each expansion in diet breadth potentially increases exposure to toxin-carrying species. Within the toxic food web, the grouper represents accumulation by dominance. It is not chemically aggressive, nor does it borrow or manufacture toxins. It absorbs them through its role as an efficient ambush predator. The reef's energy flows upward into its body, and with that energy travels chemical residue from the smallest organisms at the base of the system. The grouper becomes a living archive of what the reef has produced and transferred over time. As twilight descends and the reef's diurnal rhythm shifts, the grouper remains stationed at its cave entrance, silhouette dark against fading light. Small fish retreat into coral branches, and nocturnal hunters begin to stir. The grouper's eyes adjust to dim conditions, and it prepares for another night of ambush. Each successful strike will add to its mass, its authority, and perhaps to its chemical burden. It hunts as it always has, unaware that beyond the reef, humans may regard a fish of its size with cautious respect. In the end, the grouper's story within the toxic food web underscores a broader truth. Toxins need not be visible or intentional to shape ecological and human outcomes. A reef boss that feeds efficiently for decades can become chemically loaded by virtue of its success. The larger and older it grows, the more it embodies the cumulative history of its habitat. In clear tropical waters, it remains a symbol of strength and stillness. Beneath that calm exterior, however, lies a reminder that even the most formidable predators are part of a chemical chain that begins at the microscopic scale and rises quietly through time. At the edge of a coral overhang, where light fractures into shifting ribbons and the reef wall breaks into a labyrinth of crevices, a moray eel watches from within its narrow domain. Only the head is visible at first, a wedge-shaped skull framed by a permanent gape. The mouth opens and closes rhythmically, not in threat, but in ventilation, drawing water across gills that sit behind the head rather than beneath an operculum like most fish. The teeth are slender, recurved, built not for crushing, but for gripping. The eel's body disappears backward into darkness, its length coiled through chambers carved by time and coral growth. It is not a drifter in open water, nor a hunter that advertises itself. It is a resident of tight spaces, a predator whose geography shapes its danger. Moray eels are not venomous in the way a sea snake is, nor do they inject toxins like a box jellyfish. Their reputation for danger arises from behavior, anatomy, and occasionally from the chemical history of the reef itself. Living almost exclusively within crevices, caves, and coral hollows, morays feed on reef fish, crustaceans, and cephalopods that venture too close to their lairs. Their world is constricted. They do not chase prey across open expanses. They ambush from concealment, lunging forward with sudden acceleration and then retreating just as quickly, dragging captured prey back into the maze. The bite itself is formidable. Morays possess not only a primary set of jaws, but a secondary set of pharyngeal jaws located in the throat. When prey is seized, these inner jaws extend forward, grasp the victim, and pull it deeper into the esophagus. This adaptation compensates for the eel's elongated body and limited suction capability. In the confined geometry of a crevice where water cannot be displaced easily, suction feeding is less effective. Instead, the moray clamps, secures, and retracts. The result is a feeding mechanism that is both efficient and difficult to interrupt. From a human perspective, the risk associated with moray eels is often misunderstood. They do not seek confrontation. Most bites occur when a hand is placed into a hole without visibility, or when a diver offers food and the eel misjudges distance. The immediate danger is mechanical. A moray's teeth are sharp and backward curving, designed to prevent prey from escaping once grasped. When the eel bites, it does not simply nip and release. It clamps and often attempts to retreat into its crevice, pulling the object in its jaws with it. In the context of a human hand or finger, the danger is not merely puncture, but the combined force of bite and backward drag against rigid coral. The environment amplifies this risk. A bite delivered in open water might be manageable. A bite delivered at the entrance of a reef hole, where the eel's instinct is to withdraw deeper, becomes more severe. The coral edges are abrasive and sharp, increasing tissue damage as the eel pulls. The phrase that defines this interaction is simple. The danger is not just the bite, but where the bite happens. Within the architecture of the reef, the moray holds advantage. Morays are not venomous, yet in certain reef systems they can become part of toxin accumulation pathways similar to those described for other large predators. By feeding extensively on reef fish that may carry low levels of ciguatera toxins, morays can accumulate these compounds in their tissues. As with groupers and barracudas, the eel does not produce toxin itself. It integrates the chemical story of its prey. In regions where dinoflagellate blooms increase toxin availability at the base of the food chain, morays that feed consistently within affected territories may become chemically loaded over time. Unlike fast-roaming predators, morays are highly site-faithful. An individual may inhabit the same crevice or cave system for extended periods, defending it from conspecifics. This residency means that the eel's toxin exposure reflects the local reef's ecological state. If the surrounding fish community has been feeding on algae, hosting toxin-producing microorganisms, the moray may accumulate these toxins gradually. The eel's diet is diverse within its structural confines, but it is tightly linked to the immediate environment. Physiologically, moray eels are adapted to life in crevices. Their bodies lack pectoral fins, reducing obstruction as they navigate narrow spaces. The skin is thick and coated in mucus rather than scales, protecting against abrasion as they press against coral walls. This mucus layer also contributes to the high incidence of infection associated with moray bites. The eel's mouth harbors bacteria common in marine environments, and when teeth puncture human skin, those bacteria can be introduced into wounds. The risk is not venom, but microbial contamination combined with mechanical trauma. Predator-prey interactions around morays often involve cooperative behavior. In some reef systems, groupers and morays have been observed hunting together. The grouper hovers near the reef face while the moray probes interior cavities. If prey attempt to escape the crevice to avoid the eel, they may encounter the waiting grouper. Conversely, fish fleeing the open water predator may retreat into a hole only to face the moray within. This partnership illustrates how structural specialists and open water ambushers can synchronize hunting efforts without formal communication. The reef's architecture becomes a shared hunting ground where species exploit complementary strategies. Thermoregulation in morays follows the ectothermic pattern typical of reef fish. Their metabolic rate rises with water temperature, potentially increasing feeding frequency in warmer conditions. However, higher temperatures can also stress reef systems, influencing the abundance and distribution of prey species. Changes in fish community composition may alter the eel's diet, indirectly affecting potential toxin accumulation. Thus, environmental shifts ripple through the eel's biology in ways that are not immediately visible. The sensory world of a moray is attuned to confined spaces. Vision is relatively limited compared to pelagic predators, but olfaction is acute. The eel's tubular nostrils sample chemical cues in the water, guiding it toward injured or unsuspecting prey within the reef's shadows. Lateral line sensors detect vibrations transmitted through rock and water, alerting the eel to movement even when visibility is low. In the darkness of its crevice, the moray operates less by sight and more by scent and vibration, a hunter shaped by the tactile and chemical complexity of its habitat. From a human vantage point, the moray's reputation often precedes it. Divers encountering a head protruding from coral may interpret the open mouth as aggression. In reality, the rhythmic gape reflects respiration. The eel's caution and territoriality can appear hostile, but it is rooted in defense of a limited and valuable space. The reef hole is shelter, feeding ground, and refuge from larger predators such as sharks. To leave it unnecessarily would expose the eel to greater risk. Within the toxic food web, the moray represents a convergence of behavioral and environmental danger rather than intrinsic venom. It is not armed with injected toxin, yet it may carry chemical burdens acquired through diet in certain regions. It is not overtly aggressive, yet its bite, combined with habitat geometry, can escalate quickly. The true risk lies in interaction within confined spaces where the eel holds structural advantage. As dusk settles over the reef and diurnal fish retreat into branching corals, the moray emerges further from its hole, body undulating slowly along the reef face. Its skin reflects subtle patterns under fading light, blending with shadow. A small fish ventures too near the cave entrance and disappears in a flash of movement, pulled inward beyond sight. The eel withdraws again, jaws working methodically within darkness. The reef is a landscape of edges and thresholds, where caves meet open water and predators define invisible boundaries. The moray eel inhabits these thresholds with precision, its danger shaped by location as much as by anatomy. It is a reminder that toxicity in the reef does not always announce itself through bright colors or stings. Sometimes it resides in the quiet accumulation of diet, or in the grip of teeth within stone. The bite is only part of the story. The setting completes it. On the outer slope of a coral reef, where the wall falls away into open blue and the surge presses steadily against buttresses of limestone, a massive figure glides into view with slow, deliberate confidence. The Napoleon fish, also known as the humphead wrasser, moves not with the urgency of a hunter in pursuit, but with the quiet authority of an animal that rarely needs to rush. Its forehead rises in a thickened dome above intelligent eyes, and its lips are heavy and fleshy, framing a mouth built not for slicing, but for crushing. The scales shimmer in gradients of turquoise, jade, and pale cobalt, shifting tone as sunlight filters through the water column. It is a fish that seems carved from the reef itself, immense and self-assured, the embodiment of size and longevity in an ecosystem defined by competition. The Napoleon fish feeds with a method that reflects both power and patience. It patrols coral heads and rubble fields, probing crevices with its snout and testing the substrate with deliberate bites. Beneath those thick lips lie strong pharyngeal teeth capable of crushing hard-shelled prey. Sea urchins, crustaceans, and mollusks are not obstacles, but meals waiting to be dismantled. When it encounters a sea urchin bristling with spines, it positions its jaws carefully and applies force until the test cracks, extracting the soft interior with measured efficiency. In this act, it influences reef dynamics directly. By consuming urchins, it can help regulate grazing pressure on coral, preventing overbrowsing that might otherwise reduce coral cover. Its size confers ecological leverage. A mature Napoleon fish can reach lengths that dwarf most reef inhabitants, and it may live for decades. Growth is slow, and individuals transition from female to male as they age, a process known as protogynous hermaphroditism. This life history strategy ensures that larger, dominant males arise from the population over time, controlling breeding territories on specific reef slopes. Spawning often occurs in predictable aggregations, with individuals returning seasonally to favored sites. The longevity and fidelity to habitat tie the Napoleon fish deeply to the chemical and biological rhythms of its reef. Yet within this narrative of crushing jaws and reef stewardship lies a quieter twist. The Napoleon fish is not venomous. It does not sting or inject toxins. However, like other large reef predators, it can become part of the ciguatera conversation in certain regions. Its diet includes reef fish and invertebrates that may carry low levels of ciguatoxins acquired from dinoflagellates at the base of the food web. As with groupers and barracudas, the accumulation is gradual and trophic. The Napoleon fish, by virtue of its size, longevity, and diverse diet, can integrate the reef's chemical history into its own tissues. The process begins invisibly, among microscopic algae colonized by toxin-producing organisms. Herbivorous fish ingest these cells while grazing. Small carnivores consume the grazers, and the toxin moves upward incrementally. The Napoleon fish, feeding across trophic levels, may consume both invertebrates and smaller fish that have already incorporated toxin. Each meal adds a trace. Over years and decades, these traces can accumulate. The fish itself remains unaffected, its physiology accommodating the compounds without overt impairment. It continues to forage, reproduce, and patrol as it always has. What makes this particularly compelling is the contrast between appearance and chemical potential. The Napoleon fish looks formidable in purely mechanical terms. Its jaws can crush armored prey. Its bulk discourages most predators. Its face, with that prominent forehead and steady gaze, seems to announce solidity and strength. There is nothing about its form that suggests hidden toxicity. And yet, in certain reef systems where ciguatera prevalence is high, consuming such a fish can carry risk. The toxin is not neutralized by cooking and does not alter the fish's taste or smell. It resides quietly within muscle, the cumulative result of years spent feeding within a chemically dynamic ecosystem. Thermoregulation and environmental change intersect with this story in subtle ways. Warmer waters can promote the growth of dinoflagellates, particularly in disturbed reef environments where coral loss allows algal cover to expand. Storms, runoff, and bleaching events may create conditions favorable for toxin-producing species. A Napoleon fish feeding in such a context may incorporate higher toxin loads than one inhabiting a more stable reef. Migration patterns, though often localized, can expose individuals to different microhabitats within a reef system, each with varying toxin prevalence. The Napoleon fish's feeding behavior also illustrates the interplay between mechanical and chemical aspects of the reef. By crushing urchins and other hard prey, it alters the structure of benthic communities. Reduced urchin populations can allow coral recruits to establish more successfully, influencing reef resilience. In this sense, the fish contributes to shaping the very environment that determines toxin dynamics. A healthier coral-dominated reef may support different algal communities than a degraded one, indirectly affecting dinoflagellate abundance. The predator's ecological role and its potential chemical burden are thus linked through feedback loops. Conservation adds another dimension. The Napoleon fish is considered vulnerable in many regions due to overfishing and habitat degradation. Its large size and charismatic appearance make it a target for fisheries and the live reef fish trade. Rarity increases value, and value increases demand. In some markets, the fish is prized precisely because of its size and perceived prestige. This demand can drive risk behaviors, encouraging capture of large individuals that may also be more likely to carry accumulated toxins. The irony is stark. A species already pressured by exploitation may also carry hidden chemical risk when harvested from certain reefs. Divers encountering a Napoleon fish often describe the experience as memorable. The fish approaches with curiosity rather than fear, maintaining eye contact as it circles slowly. Its thick lips and domed forehead give it an almost expressive appearance, enhancing its reputation as a gentle giant. That gentleness is behavioral, not chemical. It does not attack divers and rarely displays aggression. Its power is reserved for prey that cannot outswim its reach. The human threat arises not from the fish's intent, but from the complex chemistry of the reef it inhabits. Within the broader narrative of the toxic food web, the Napoleon fish represents the culmination of upward movement. It is a top-tier consumer that integrates energy and matter from multiple levels below. The ocean's chemistry does not remain confined to microscopic organisms or small grazers. It ascends, passing from algae to fish to apex predator. The Napoleon fish, massive and serene, becomes a living testament to this ascent. Its body contains not only muscle and bone, but traces of countless interactions that began at the smallest scale. As the sun sinks toward the horizon and shafts of light angle across the reef, the Napoleon fish turns slowly, vanishing into the blue beyond the coral ridge. Smaller fish scatter briefly in its wake, then resume their routines. The reef hums with life, a tapestry of teeth, spines, and stings. Yet beneath that visible array of defenses lies a subtler force. Chemistry flows upward through feeding relationships, shaping risk and resilience in ways that cannot be seen at a glance. The ocean is not only a theater of physical predation, it is also a conduit for invisible compounds that move from microbe to megafauna. The Napoleon fish, rare and immense, stands at the intersection of power and accumulation. Its crushing bite defines its ecological presence, but its chemical story reflects the reef's deeper currents. In the end, the danger in the sea is not just in teeth or venomous spines. It is in the quiet ascent of toxins through time, carried upward by the very success of those who feed at the top.